Voilà.
Essaye.
So Sylvia, can we start?
Yes, whenever you want.
So I can share my screen. Okay. Bonjour à tous. Good morning to everyone and welcome to the second Biomedical Engineering Symposium. This is an IP Paris INRIA joint event, and it is held
Désolé, on a des petits soucis. I'm sorry, we have some trouble with connection with Abdul. We are trying to solve it soon.
Et mon fichier, il est sur le bureau. Je sais pas comment tu le partages.
Si on peut l'ouvrir déjà.
Il est sur le bureau, mais où?
Ça s'appelle BMI Symposium. Symposium PME. Oui. Donc là, je vais pour revenir à Zoom.
Si ça fait.
Là.
Là?
Ouais.
Ouais. So.
Donc, ici, je vais. Écran.
C'est bon?
C'est que je fais les présentations.
Vous êtes en train de c'est bon?
Oui, mais c'est pas en mode présentation. C'est peut-être là. Disponibilité.
Probably, Abdul, you can try with lire.
With where?
Lire, so on the right, close to Keynote, above the X and S logo. Gauche. Voilà.
Here?
Lire. So where there is the play symbol. On the right. There.
Arrêtez vidéo?
Non. Yeah. Vous avez juste lancé, c'est ça?
Mais je vais passer en presentation mode. Édition.
Non, attends, c'est pas. Lancé.
D'accord. Ok.
C'est bon?
Je pense. Yes. Okay. I am really sorry. I had a Wi-Fi connection that I thought was going to be fine and then ended up not being fine. Can you hear me okay now?
Yes.
Okay. So let's start over. I'd like to welcome you all to the second Biomedical Engineering Symposium. This is a joint event, IP Paris INRIA, and it is held through the generous support of the Fondation Bettencourt-Scheller. This is the second BME Symposium. The first one was actually held in December 2019. And this is an annual event, so it was intended, this second one was intended initially to be in December 2020, but because of COVID, we ended up getting delayed by about 6 months. So for those of you who don't know me, I'm Abdul Barakat. I'm a CNRS Director of Research in the Hydrodynamics Laboratory at École Polytechnique. I'm also a professor at École Polytechnique. This symposium is, like I said, a joint event IP Paris INRIA. So let's just say a few words about biomedical engineering. So biomedical engineering, as most of you know, is basically the application of physical sciences and engineering to disease, right? To understanding, diagnosing, and treating disease. And at IP Paris, our biomedical engineering program has various components, so various subfields, if you will. Some of our researchers are specialized in biophysics and biomechanics, and this happens at multiple scales. Some of us are studying things at the microscopic, molecular, cellular scale, and then some at larger scales. We have a certain number of colleagues who are interested in imaging, and they're typically experts in signal and image processing, as well as developing imaging systems. Sensors and devices is another sub-track of our biomedical engineering program. Then we have people who are studying biomaterials and bioinformatics, and you'll hear some of these various subfields represented in different presentations that we'll hear today. Before getting started in the program, I'd like to kind of acknowledge our biomedical engineering steering committee. So these are people who are very involved in the whole program of biomedical engineering, and they've given a lot of their time and effort, and you'll meet some of them today. So Anne-Chantal Gouget is a chemist by training who's in the condensed matter physics laboratory. You'll meet Anne-Chantal later today. François H is the director of the LOB, the Laboratory of Optics and Bioscience, and you'll also meet François later today. Dominique Chapelle is an applied mathematician in INRIA and LMS, the Solid Mechanics Laboratory, and he's part of the steering committee. So is Thomas Simonson, who is a computational biologist at the biochemistry lab. Marc Lavielle is an applied mathematician, joined INRIA and applied math lab. Angelo Pierangelo is a physicist who is interested in imaging systems and is part of the PICM, so the physics of interfaces and thin surfaces lab. Sylvia Corsini did most of the work really preparing for this symposium. Thank you very much, Sylvia. So Sylvia is our BME manager. She's also a neuroscientist on the side, and myself. So this is our BME steering committee. I mentioned the generous support of the Fondation Bettencourt-Scheller. So another thing, in addition to the symposium, that the Fondation Bettencourt-Scheller is currently funding in our program is a BME seed grant program. And so I'll talk a little more about these seed grants in a minute. But basically, the vision behind this program is that there's a convergence of a number of technologies that are being developed by physicists and engineers that are going to be very, very important, and that are, in fact, very, very important for medicine, right? So what we today see is that medicine is becoming, as a result of these technologies, a lot more personalized, a lot more targeted, and a lot more connected, right? So if you think of flexible electronics and the ability to make, for example, sensors that can be implanted on curved surfaces, if you think of machine learning and the ability to acquire a lot of data and then learn from it, if you think of bioprinting and the ability to basically print tissues and cells, if you think of very fast computing, if you think of nanoscience and nanotechnology, where we can now have very small scale sensors and biosensors, and if you think, of course, of connected devices, and you put all of these elements together, and you really have the makings of a revolution in medicine. So we feel it's very important to have close collaborations between physicists and engineers who are developing these technologies and clinicians who are going to be deploying them and using them to hopefully improve patients' lives. And we need to do this in the right way, right? We need to do it in an ethical manner. We need to do it in a responsible fashion. So that's kind of the vision of the seed grant program that the Fondation Bettencourt-Schiller is funding. And basically, the program involves a total of 11 seed grants that are awarded competitively over a period of 5 years, which is the life of the program. And the idea is each seed grant is a collaboration between a BME researcher at IP Paris or University of Paris-Saclay in collaboration with a medical doctor. So each one of these seed grants is a 2-year grant. It typically provides funds to hire a postdoc and cover basic lab fees. The projects are selected by an external scientific board. And what's basically taken into account in the evaluation is the scientific excellence of the project, as well as the potential for socioeconomic impact. Yes, as I mentioned, the funding decisions are made by an external international scientific council whose composition you see here. Some of these colleagues are probably here already, and some of them will be joining us later on. For those in the US, it's a bit early right now, since about 3 in the morning on the East Coast. So I'd like to acknowledge our scientific council nevertheless. So Ross Ethier is a professor of biomechanics at Georgia Tech. Irene J.R. Jokudi is at Tufts University, a professor there, a specialist in imaging and in imaging systems. Catherine Picard is an expert in biomaterials and tissue engineering at Grenoble. David Saloner is at UC San Francisco, an expert in imaging, image analysis in concert with biomechanics. Cécile Sykes is actually, this is an older slide, says Institut Curie. Actually, Cécile has moved to ENS, Paris, and she is an expert in cellular biophysics. And then Michael Tanter, who's at ESPCI in Paris, who's an expert in ultrasonic imaging and specifically elastography. We have a very exciting program for you today. We're going to start with my welcome comments, and I'm already behind on that. I really apologize with my connection problems. So we're going to first start with 5 presentations from colleagues at IP Paris in different aspects of biomedical engineering, and they come from the different establishments, the different institutions that make up IP Paris, just to give you a flavor of kind of the range of activities and research that's going on in BME at IP Paris. So we'll start with that. Then we'll have a little break of 15 minutes around 11 o'clock. Then the second morning session is going to be a focus on some of our young researchers, some of our PhD students and postdoctoral fellows. That session will be chaired by Anne-Chantal Gouget. Then we'll break for lunch for an hour. We'll meet up again at 2 to basically announce the laureates of the 2020 cycle of the Biomedical Engineering Seed Grant, which again is funded by the Fondation Bettencourt-Schiller. And that session will be chaired by François H, and the laureates will give brief presentations. In the afternoon, we have some institutional presentations. Eric Labaille is the president of École Polytechnique. He intended to be here, but some last minute thing came up. So he still was really very interested in making a presentation. So we recorded a presentation for us that we will show. Armand de Boissière is the Secretary General of the Fondation Bettencourt-Schiller. He'll give us a presentation as well. And I think Armand and Eric, the order has been reversed in the final program. And then we'll end the day with a plenary lecture by Professor Alexandre Mebaza, who is a a surgeon and a medical doctor at the Université de Paris. And I think having a medical perspective would be kind of a very apropos way to try and finalize the day. And then we'll just conclude. So I hope you'll enjoy the day. Thank you very much for being here. I think with that, I'll stop. I'll just mention just one thing. So I actually had a death in the family. My cousin passed away from COVID complications the day before yesterday, and the funeral is actually today. So I will have to leave you at right after the first break. No, sorry, I'll have to leave right around lunch time, and to go attend the funeral. So I apologize for that. But Francois Hache has generously agreed to take over in the afternoon and handle everything, and he'll do a much better job than I would anyway. So with that, I'll stop. And we'll start now the next sessions. Let's see. So maybe I'll start by introducing our first speaker of the morning session. And the first speaker is Jean Boisson, who is from the Unité de Mécanique at ENSTA. And he's going to be telling us about mandibular distraction, osteogenesis properties, and evolution. So these, Jean, you've got 10 minutes for your presentation, and then we'll do a few minutes of questions.
Okay. Thank you, Abdul, for the introduction, and hello to everyone, and thank you for listening to my presentation on mandibular distraction osteogenesis. So I'm Jean Boisson. I'm working at ENSTA, and this is a project I developed with Natasha Kadlu, which is a maxillofacial surgeon in Necker Hospital. So the first question is, what is mandibular distraction osteogenesis? So it's a surgical procedure that leads to do progressive lengthening of the mandible. It's a general procedure. It can be done of every long bone, but here we are focused on the mandible. And the indication for that is skeletal deformities and reconstruction, face reconstruction. And it's generally a children's surgery. Here you have a picture of a little girl before the surgery and after the surgery, and you see that the mandible is quite small before and quite regular after. And so the principle of this surgery is to use the property of the periosteum to reconstruct the bone. The periosteum is a tissue that is anchored on the bone. And so the procedure is like that. You have first in the OR to do an osteotomy, which is a rupture or fracture of the bone, and you implant a distractor. A distractor is this dispositive, this device, and it's composed with two plates and then the screw. And so when you're rotating the endless screw, the two plates will go apart, and so as the bones attached to them. So the procedure, when you do that in the OR, you wait for 5 days and wait for stem cell recruitment. And after that, for 20 days or 30 days, depending of the length you want to gain, you have to do an activation, and it's done by the parents. So they have to rotate this rod for 1 millimeter per day. And after reaching the good length, the surgeon remove the rod, and after 5 months of consolidation, the distractor is removed. So it's a very invasive procedure, and it's quite new. I mean, it started in the 50s, but it's coming to be in routine in the 90s. But there's still some problem with this surgery, and especially with the transmucosal and transcutaneous activation rod, as you can see here in the picture, because it causes infection, scars, pain, and occlusion problem. So we came with an idea to replace this rod with a magnetic transmission between two permanent magnets, one internal, which is connected to the endless screw, and one external, which is bigger. And when you rotate the external magnet, you apply a mechanical torque on the internal magnet. And if this torque is high enough, you can activate the distraction. And this way, you don't have any complication due to the external rod. So we came with that idea, and we wanted to know if it's possible. So we wanted to do a distractor sizing. And the first question was, what is the load applied by biotissue on the distractor? We did a quick review, and we only find one paper by Robinson in 98. And they found that the force is about 38 Newton. So we wanted to confirm this value, and we wanted to test our hypothesis that the periosteum is the principal tissue opposing the DO, destruction of osteogenesis. Just to come on the periosteum, so is the conjunctive tissue which is anchored on the bone and which is charged to regenerate and nourish the bone. To test these two hypotheses, we did two campaigns of measure, one global on cadaveric subject and one on samples. What we do is very simple. We measure the torque applied on the external rod for a 20 millimeter DO, and we get the force per millimeter in this way. And the other end, we get two samples of the periosteum, one vestibular and one lingual, and we did a uniaxial tensile test along the DO direction. And thus we can compare the two force to stress coming from the two procedures. What we got from the cadaveric study is a load coming from 18 to 50, which is confirmed what's found Robinson. And from the tensile test, we got typical curves for conjunctive tissue with a small stiffness part and a high stiffness part. And so we evaluate the force coming from the periosteum by calculating the deformation in the high stiffness part, because there is some hypothesis that the periosteum is already pre-stretched on the bone. And so when we do this calculation, we got a stress of a load of 12 Newton coming from the periosteum for a 1 millimeter deformation. Elongation, and 80 compared to the 18 we found in the cadaveric study. So it helps us to evaluate the force that we need to do to apply with the distractor, with the magnetic distractor, and it's confirmed what happened that the periosteum is the principal tissue opposing the DO. And with that, we could evaluate if we can activate the distraction by magnetic magnet, by permanent magnet. And we found that with the current sizing we want to use for the magnets and for regular friction, we are able to reach the 50 Newton we want. So this is fine, and we developed the Vivo Dogma project from there, and it's the 2 years project that helped us to develop a lot of prototype of distractor, a lot of activators. You have pictures here, there, and there. And we started 3 negotiations with third-party operator, everything with the help of SAT Paris-Saclay. And so we are reaching now the final product that we want to transfer to a third party. Just to prevent you, they will show you a little movie of an activation of a distractor on a cadaveric subject. So be careful, it's just 30 seconds, 20 seconds, so I will stop right here. So it's the first prototype with the first prototype of activation, so which is basically electric screwdriver. And you see that in the bone, the two parts of the bone are going upwards with this activation. We improve this device right now, and now we are quite confident that we have a final product during this summer. And this development helped us to find a new academic research area. First, for example, on the periosteum, that we wanted to study the structure and to compare the structure with what we found in the tensile test. And using a structural approach coming from Cominu and Manshot, we compare the structural model with what we got in the tensile test. And we found that taking the parameter of the structure from histologic pictures, we can reproduce satisfactory production, of what we got in the tensile test, except for one thing, which is the fibrillation amplitude, but this coming from the periosteum shrinking when it's not on the bone, as it is pre-stretched on the bone, it shrinks when it's not, and so we get lost of the amplitude of the collagenic fiber. Just to finish with the perspective we got from there, we wanted to develop two kind of thematics, one medical and one more academic. We want to develop our magnetic activation system on different devices, on magnetic palatine disjunction, even on lateral periosteum distraction, which is a collaboration with George Cardona, which correspond to get more thickness on the mandible. And from the academic era, I will not go into the detail, but a project that is very interesting is to study the influence of the static magnetic field on the bone remodeling, because this was studied but just on the cellular level, and what we got, we want to see if they can see the influence of a magnetic field on the bone itself and on the mechanical response on the bone. And this will be probably a subject of a PhD in 22. Thank you for your attention.
Thank you very much, Jean, for this very nice presentation that I think illustrates the translational potential of some of our work. Okay, so the Jean's presentation is now open for questions, and questions you could either type them in the the Q&A window, or you can raise your hand and ask the question directly. So let's see. I see one question already from Emmanuel Borter. Emmanuel says, interesting, wondering how painful are these procedures, the standard ones?
The standard ones are quite painful, because you have a road going through the skin or the cheek by inside, and basically you rotate some mechanical parts in the flesh directly. It's kind of painful, and moreover, it's as it's the parents that have to do that. You basically torture your own child, which is very difficult, and can be even a problem, because as it's a very difficult moment, the parents are can do nothing good, because they can turn in the wrong sense. For example, generally you do this procedure in the both mandible parts, and so time to time it's happening that the parents are turning in one sense for this part and the other sense for the other parts, and so you have a mandible which is not in the good vector anymore.
Thank you. I have a question, actually. So how do you anticipate moving this to the clinic, or what is going to be the path to get this all the way to the patient?
I mean, the trajectory you want to know? Yeah. The trajectory for the moment, it's kind of just surgical I would say, experience. The surgeon knows that for this kind of deformities, you have to do more this way or more this way. But it's kind of something interesting to calculate the right vector, because even more, you can have this procedure not just for the mandible, but for the whole face. And when you have this procedure for the whole face, you have 4 distractors. And then you have to calculate the right vector, knowing that you have these 4 distractors to change and this is not calculated for the moment, and it's just the surgeon saying, okay, do 2 turns here, 1 turn there, half a turn there, and this could be dramatically more studies.
Great. Thank you very much, Jean. I think
Thank you.
with that, we'll move on to the next talk.
Thank you very much.
So the next talk is by Cataline Fetita from the Samovar Laboratory at Télécom Sud Paris. And he's going to tell us about quantitative follow-up of pulmonary diseases. So Cataline, welcome, and the floor is yours.
Okay, do you hear me?
Yes, perfectly.
Thank you for the introduction, and thank you for the invitation to the seminar. I'm glad to share with you this short overview of activities in the Biomedical platform of Télécom Sud Paris with a focus on pulmonary diseases. So Biomedical stands for Biomedical Imaging and Clinical Applications. It is a physical platform where we try to develop solutions centered on pathologies and answering clinical needs. In this talk, I shall focus on applications in lungs, in pulmonary applications, covering respiratory and infiltrative lung diseases. So in all these cases, the objective is starting from clinical imaging modalities, in occurrence the X-ray CT scans, to extract image biomarkers able to diagnose, quantify, and monitor disease progression over time. So concerning the respiratory pathologies, we are focusing on asthma and chronic obstructive pulmonary diseases, which are both responsible for airway remodeling with a partially reversible airflow reduction in asthma and alveolar space destruction in COPD, leading to emphysema. So in these cases, image biomarkers rely on the segmentation of the tracheobronchial tree from volumetric CT images, as you can see here in this 3D reconstruction. And on the right-hand side, you see the correspondence on the actual CT scans images with the airway lumen blinking in red. So what we reconstruct here is the airway lumen. So from the 3D morphology of the airway tree, of the airway lumen, we are now able to detect pathological variations of the caliber, the lumen caliber, caliber shown here in false colors. So we aim for shrinking or dilations known as stenosis and bronchiectasis, and also depict them, their spatial location on the axis, medial axis of the 3D structures. Moreover, the quantification of bronchial remodeling, targeting in this case both lumen and wall, can be performed either in transverse sections, as you can see here, along any path computed from the airway axis, or directly in the 3D space. So these 2 biomarkers related to the airway lumen and wall have been applied in several clinical studies on moderate and severe asthma and have been included in a computer-aided diagnosis platform implemented on clinical site, actually. Additional applications in 3D augmented visualization and navigations from external side or from endoluminal analysis have also been proposed. So here the augmented information in this video is the airway caliber, but other kind of information like airway wall can be also displayed. Other possible applications are in interventional planning with guided navigation. In this example, I show a simulation of a transbronchial biopsy of a nodule. The nodules are shown here in yellow, where we are guiding the virtual camera from the trachea through the target point through this structure to perform the biopsy non-invasively or minimally invasively. This path, this trajectory, it's computed automatically on the airway axis. So other applications in guided intervention, guided navigation could be related to stent pause or bronchial thermoplasty. So airway biomarkers can also be associated with vascular information. In this example, the degree of the airway wall thickening together with the ratio, the caliber ratio between the airway lumen and the homologous artery are able to depict the therapy response in bronchial thermoplasty as shown here. So different kind of responses can be expected from status degrading to status improving, which suggests that patient stratification is a key issue for this kind of therapy. Finally, functional prediction or functional behavior, sorry, for related to a given airway morphology can be predicted based on patient-specific airflow simulations, as shown here, where you can see the airway pressure on the walls and airway flow velocity in a respiratory dynamic regime simulated. So let's jump to another class of pathologies, which are infiltrative lung diseases, where we focus here on COPD and fibrosing idiopathic interstitial pneumonia, both responsible for an irreversible process of lung function decay and requiring regular quantitative follow-up with the CT for assessing lung lesion progression. In this case, the classification of different lung lesions is addressed with a deep learning model able to differentiate between normal tissue, depicted here in green, emphysema regions in blue, and fibrosis and ground glands consolidated in a single class called IEP in pink. So this allows to perform a quantification and a follow-up for the entire lung volume, as you can see here, for a patient monitored at 1 year interval for two years. Complementary information for disease phenotyping beyond texture classifications can also exploit information on vascular remodeling, in this case being related to the pruning of small caliber vessels, and also the presence of traction bronchiectasis, again dilations, related to fibrosis, and related to the, or induced by the mechanical stress of the respiration in the presence of the fibrosis. In this case, the biomarkers related to traction bronchiectasis also demonstrated a predictive power and showing a good correlation with the follow-up pulmonary function test. So another pathology that we experienced recently, and unfortunately, is COVID-19, which is an alveolar interstitial pneumonia suspected to evolve to FIP in 10 to 30% of severe cases. So this means that it is important to, again, to follow up the lung disease progression in this case. So for this, we have developed a similar model as for FIP, for lung tissue classification, but this time separating between fibrosis in pink and ground glass regions in light blue. In this example, you see a COVID moderate case showing a short time recovery where the lung pathology or lung disease disappears almost completely after 6 months. And in the second example, we have a severe cases with a long time recovery, and we see the persistence of sequela in terms of ground glass opacities after 9 months of follow-up. So COVID-19, it's also an acute vascular lung disease. So we are investigating currently this direction, looking to assess the vascular remodeling in terms, again, of pruning of small caliber vessel, estimating the pruning of small caliber vessels with adapted biomarker, in this case, depending or defined by the volume, blood volume ratio of small vessels versus all vessels. And preliminary results seems confirming there is a high correlation with the severity of the disease of this biomarker. Finally, we are also interested in looking for biomarkers extracted from lung dynamics assessed with inspiratory versus expiratory CT scans. So we are looking here at biomarkers related to lung deformation. So here you see the lung deformation amplitude for different FIP or COVID patients and one control. And also we look at image biomarkers able to characterize or detect the inconsistencies or abnormalities between inspiration to expiration. And ultimately, we are currently collaborate with the medicine team of ex-INRIA to perform a patient-specific lung mechanics simulations aiming to predict lung degrading in fibrosis due to mechanical traction effects. And more detail on this aspect will be given in the next presentation by the next speaker. So this allows me to conclude my presentation, and thank you for your attention. Thank you.
Thank you very much for this very nice presentation. The presentation is now open for questions. Again, you can ask questions either by typing them in during the presentation in the question and answer window, or by raising your hand and we'll call on you. Actually, I have a question. So in the beautiful movies that you showed where you were navigating down the airways, how far down can you go? Like what generation?
Well, I think it's 7th, 8th generation. Everything will depends on the segmentation accuracy, and also it depends also on the CT scan. So in this example, so we are going quite far below in the segmentation. So everything will depends on the CT scan and how far we can go with the CT reconstruction. But globally, we are, I think, on around 7th generation.
Okay. Any other questions for Katalin? I have maybe another one real quickly. So you mentioned the idea of kind of detecting stenoses and or enlargements in the caliber, right? So I assume there are kind of natural variations in the diameter that happen even without pathology. So how do you discriminate kind of between what is a pathological stenosis and what is just part of the normal,
Yeah. So. Indeed, there might be some variations for in the caliber which are normal, namely in the bifurcations, and this can be seen with the double analysis along the airway axis. So when you look at perform this analysis by navigating in a way on the airway axis, and also looking at the global caliber that you can see depicted here, and which is computed using a granulometric approach. So it's not only the distance from the axis to the wall. And this information coupled with also a kind of sensitivity factor gives us the possibility to tune up a little bit the sensitivity of the detection. So of course the objective is to have a low rate of small positives, of course, and not missing these variations.
Okay. Okay, if there are no other questions, I think we'll move on. Thank you very much.
Thank you.
Okay, as has already been alluded to, the next speaker is Martin Genet, who is part of the joint INRIA LMS medicine team. Martin is also on the faculty of mechanics at École Polytechnique, and he's going to tell us more about the pulmonary system and personalized pulmonary poromechanics in health and disease. Thank you, Martin.
Thank you, Abdul. So can you hear me well?
Yes.
Good, good. Yeah, so thank you. Thank you also to the committee for giving me the opportunity to present some of my work today. Congrats also on the organization. I think having me speaking right after Katalin is probably one of the smoothest transition I've ever seen in a scientific workshop. Indeed, I'll be talking now about the work that we do to combine the image processing that Katalin does and our biomechanical modeling to address some questions in pulmonology. So this is collaborative work inside Medisim, also with Katalin at Telecom and with a group of pulmonologists and radiologists at Avicen. Yeah, but before that, so for those who don't know, Medisim stands for mathematical and mechanical modeling with data interaction in simulations for Medisim. It's a joint team between École Polytechnique and INRIA. So in the team, collectively, we develop biomechanical models, also numerical methods to solve this model and basically merge the modeling and the data. Some people also do mathematical analysis of the methods. We usually implement these methods to actually solve real problems. We also develop experimental methods. So for a long time, the team was really focused on cardiovascular applications, but now over the past couple of years, we've broadened a little bit the spectrum of application to pulmonary system, the skin, the cornea, etc. And as an example, today I will focus on our work on lung modeling in the context of idiopathic pulmonary fibrosis. Yeah, so again, it's a collaborative effort. We've been funded by INRIA through the fellowship of Cécile Pat, the PhD fellowship of Cécile Pat, who actually just received the L'Oréal UNESCO Thesis Award. We also received the ANR Jeune Chercheur in 2019 and an ANR COVID last year. So the clinical context now was already mentioned a little bit by Katalin. I will focus now on what matters really to us mechanicians. So idiopathic pulmonary fibrosis is so the symptoms basically of the disease are quite well understood now. So you see, for instance, on this CT images, you see the difference between the normal lung here and a fibrotic case is quite clear. Also on the alveolar scale, we know the pathophysiology of the disease we see that the septa between the alveoli becomes much thicker. Some of them get damaged, etc. So you see that the gas exchange here are really less efficient. And we see also a signature of this disease on the mechanical response of the lung. That is usually the pulmonologists, they look at graphs of volume versus pressure. So you see a normal lung here and a fibrotic lung here. So you see the curve is below. So it means the lung is stiffer, is less compliant. But it's still a poorly understood disease. It's called idiopathic, which is a quite explicit name, at least in French, to say that basically the mechanisms by which the disease appear and progress is still largely not understood. So we are interested in this disease, as I said, because it has an impact on mechanics, but actually the real reason, and actually the reason why the doctors contacted us is also because it's also the other way around. It has been hypothesized that there is some kind of mechanical vicious cycle, such that mechanics also plays a role in the progress of the disease, such that basically when the lung is stiffer, you need to pull harder on the lung to inflate it, which induce higher stresses, which in itself induce higher fibrosis rate. So this is the fundamental question, let's say, around the disease. And better understanding these processes would allow to have a better diagnosis, even maybe prognosis, and optimizing or analyzing treatment for this disease. So that's the kind of questions that we want to address. So our approach is somehow similar to what we did on the heart, in the sense that we were developing physical modeling, numerical simulation to solve these sometimes quite complex models, and estimating parameters of the model from the data to basically derive biomarkers that can be useful for the clinics or for the understanding of the disease. So in that respect, it's important to be not too far away from the actual clinical data if you want to be able to estimate anything from the model. So here, the actual data that we can get are thoracic scans, CT scans on patients, and we're lucky in some sense because since a few years now, it's recommended to do 2 scans, to have 2 time steps, so beginning of or end of expiration and end of inspiration. So for us, it's very important because thanks to these 2 scans, we can see the breathing motion, the kinematics of breathing. Unfortunately, it's basically impossible to measure the breathing pressure in vivo in patients. It's way too invasive. So everything we do has to be relative to some kind of normal pressure that we inject in the model. We develop a model based on pore mechanics. So I will come back to this later, but pore mechanics basically is an idea that we consider the lung as a continuum, as a mixture of fluid and solid. We don't represent explicitly the microstructure of the lung, and we're going to model these 2 states of end expiration and inspiration, considered to be in some static equilibrium. Yeah, so I put there some remarks. We've been discussing a lot with the doctors over the years about many questions regarding why do we do physical modeling versus pure data analysis? Why do we do mechanical modeling versus just pure kinematical analysis? Also pore mechanics versus pure solid mechanics, etc. So I don't have time to discuss all of this now, but if you're looking for a question to ask at the end, feel free to use this. Yeah, so now I will give some details about the model, the estimation pipeline, and then go to the results. So as I said, we're using pore mechanics. So pore mechanics, it's not a French thing, but it was the foundations were definitely laid down in France, especially by Bio in the 40s, 50s, etc., and even the 70s for the finite strain framework. So here, as I said, we're considering the continuum is a mixture of different phases, fluid and solid. And so now when we're computing the deformation of a domain, on top of the usual mechanics unknowns, displacement, strain, stresses, now we have also the quantity of fluid, how the fluid moves inside the structure, and also the fluid pressure. And when we write, classically, when we write conservation equation, we're getting a system of coupled nonlinear partial differential equations to solve. This is general framework of pore mechanics, and we have to make some specific derivation for the lung. So we built material laws that are specific for the lung parenchyma. We also designed specific boundary conditions. So the lung here is surrounded by this pleura, which is made of 2 layers, the parietal pleura here and the visceral pleura there, and in between some pleural fluid. So basically, when you breathe, you pull on the diaphragm and the intercostal muscle, which will basically pull on the parietal pleura, create some kind of depression inside the pleural cavity, which will pull on the visceral pleura, which will then pull on the lungs. So we have some boundary condition such that the lung basically stays in contact with the hard part of the thorax and can be also inflated by the pleural pressure. And we also had to derive build some solutions for specific problems that we had for the lung modeling. I'm not gonna detail this now. So at the end, this is the kind of result simulation that you can get. So it's you see a lung getting inflated by some breathing pressure, and so you see one lung here, and this is the thorax, the thoracic cage. The lung stays in contact with the thorax, and here in color you see the displacement, and here the stress. Okay, so now that we have a model, as I said, we estimate some parameters of the model based on the data to derive some biomarkers. So this is where the collaboration with Katalin is key to us. So thanks to Katalin basically provides us with the segmentation of the lungs and the disease region. So from the segmentation, we can drill some finite element mesh here. So you see the mesh of the lungs and the thoracic cage. We also can project the disease state. So here we have a fibrotic region. Here we have a healthy region onto the mesh. We can also, from the images, we get the porosity, the local porosity of the tissue. We also, from the images, extract the boundary conditions, so the motion of the thoracic cage here, on which the lung has to stay in contact. And most importantly, now we estimate some material parameters based on the images. So here we use a simple derivative-free approach. So the implementation is quite simple. You start from some material parameter for the healthy and disease region. For instance, you run the forward problem. And now we're going to compare the kinematics of the model and the kinematics that we get from the images. And we're going to iterate over this to find the material parameters which are going to minimize the distance between the response of the model and the observation of the data. And this is the kind of result that we have here. So this is the compliance for a few, so one control case and 3 patients. So here these are 2 plots for just two different ways to parameterize the model, but not really important. So you see that for the healthy case, we have just one region, so it's the global compliance. And for the patients here, we have 2, you have to look at the blue and orange. So these are the compliance of the healthy region in blue and the disease region in orange. You see that the second case didn't work so well. So we have a solution for this problem, but we didn't implement it yet. So I will focus on P1 and P3 and focus on this plot here. So yeah, as I said, we can estimate the compliance of the healthy and disease region. We check that they are in the range of expected value. We see that the compliance of the disease region of the fibrotic region is lower, so it means it's stiffer as expected. And we can also characterize, compute the ratio, for instance, the stiffening of the tissue. And this marker here could be useful to better classify the patients and see the potential effect of some disease, of some treatment, sorry. So that would be the more, let's say, applicative aspects of the work. And as I said, also we're also targeting some more fundamental questions about how mechanics drives the progression of the disease. So here we can study, thanks to the model, we can study, for instance, the stress distribution inside the tissue. And we see, for instance, on this patient that you see like somehow higher stresses at the frontier, at the border between the disease and the healthy region, which is the region basically where the disease progresses. So
you want to start wrapping up? We're a little
yes, sorry. Yeah, my last slide actually. Thank you. So yeah, I presented this mechanical model of the lung at the organ spatial scale and breathing temporal scale with the personalization procedure based on clinical data. It's the first step toward quantitative analysis of potential vicious cycle in idiopathic pulmonary fibrosis. Improved in the sense of engineering, so more objective and more quantitative classification of patients, maybe a follow-up study of antifibrosing therapeutics. And we have many things going on right now in this project. We are trying to study more cases, including longitudinal data for some patients, improving the model by introducing alveolar scale, the surfactant effect. We're also trying to validate the model based on lung experiments through a collaboration with Aline Delbruno at LAMCOS. And we're also working on adapting the pipeline to MRI through a collaboration with Xavier Metz at Biomaps. And with that, I thank you for your attention.
Thank you very much, Martin. Any questions for Martin? Probably have time for 1 or 2 questions. Okay, I see there's a question in the chat. Can we? The question comes from Saeed Nala, who says, "Can we use solid interaction modeling for the same analysis? How different will it be from poromechanical modeling?"
Yeah, good question. So, yeah, right now we have only 2 data points, end exhalation and inhalation, and we somehow assume that the fluid is at equilibrium at this phases, especially because the patients need to basically take a specific breath for the imaging. But anyway, this is probably an assumption that's not always valid. So indeed, making some more actually computing the flow of air might be interesting. So it would be quite involved, especially if you want to compute the flow everywhere in the lung. I mean, it's a quite complex network of it's a quite complex network, but yeah, it might help. It might improve the pipeline in some situations, but we are not quite there yet.
Okay. I have a really quick one. So I was reading about fibrosis after COVID, and it seems that in some cases it's actually reversible. And I was wondering if your models could somehow give insight as to potential reversibility of the disease.
So that's also one of the aspects of our COVID work, is to basically see if we can find. So we're gonna study patients, right after the COVID, and after several months after in the year after the COVID, and we're gonna try to see if we find some markers maybe right after COVID that can predict which fibrosis is gonna solve itself or become problematic. So this is definitely something we want to look into.
Okay, thank you very much, Martin.
Thank you.
So the next speaker is Pietro Gori from the LTCI laboratory Telecom Paris. No biomedical engineering symposium would be complete without a presentation on machine learning. So we're going to learn about artificial intelligence and anatomical medical imaging. Pietro, the floor is yours.
Thank you. Can you see my screen?
Yes.
Maybe just put it. Okay. Okay. Now it will be better. Thank you very much for the presentation, the invitation. So yeah, as you just said, in this talk, I will present our research work about artificial intelligence and anatomical medical imaging. I would first like to present the researchers and PhD students that work on this. We are 2 researchers. Actually, Isabelle is on leave for 4 years, but we are still collaborating. We have 3 CIFRE PhD students right now and 8 academic PhD students. And we currently have research work with several clinical partners, especially Santana Hospital and Necker Hospital, and academic partners like Neurospin, MapSync, and Imbria or University of Sao Paulo. And we also have industry partners, especially through CIFRE PhD students with Philips, GE Healthcare, and Gerber right now.
So sorry, Pietro, I don't know if you've already advanced your slides, but we still see the title slide.
Oh, that's okay. I don't know why it's that's weird. Can you? I don't know what's okay. I can try to redo it because I was actually moving my slides.
Yeah, we couldn't see the.
Okay, try to do it again. Can you see now the slides?
We see a different slide, yeah.
Different slides, okay, so we're just keeping like this. So sorry about that. So this was basically the slides where I present the researchers and PhD students and the clinical academic partners and industry partners, just to yeah, not to show it again. Okay, so about the research topics that we work on, there are several of them, like especially statistical and deep learning, such as transfer learning, domain adaptation, knowledge distillation, knowledge representation, shape analysis, computational anatomy, and as clinical application, mainly segmentation, registration, atlas construction, and 3D modeling and surgery planning. But today I would like to speak more about three main topics about which we are working lately, which are modeling, how to model basically anatomical knowledge, how to transfer anatomical knowledge, and how to discover it. So the first part, we proposed basically to use either graphs or fuzzy spaces to model the prior anatomical knowledge that we might have. And this prior anatomical knowledge might be, for instance, the position of some anatomical organs. So we know that some organs are on the left or on the right. And we can model this either using some graphs or using fuzzy models. And with fuzzy models, we can also model more complicated relationship, not only special one. And we can also model something like, is it in between, or is it on the top, or is it in the curved space, and something like this. And we use these modeling representations especially for gray matter of the brain segmentation, and also for white matter of the brain segmentation. And this is work that we have already published. And another way that we use to model the anatomical prior knowledge that we might have is also by using some geometrical constraints. Here is the simplest example, I would say, are some landmarks that can be positioned by clinicians. And then we use these landmarks to basically constrain a deep learning segmentation model. And here we use it for vessel segmentations in order to produce 3D individual patients for pediatric surgery. Then the second part of the talk is more about how can we transfer the knowledge that has been learned from, for instance, from one deep learning network or from one domain to another. And the first example is if we want to, is based on the privileged information and generalized knowledge distillation framework. And the idea here is that we have a teacher network that has been learned using different modalities, in this case, 4 different modalities to segment brain tumor. And then we say, okay, the teacher network is very nice in research domain, but in the clinical domain, we usually have only one modality. And so here the idea is how can we transfer knowledge from this teacher network to this student network in order to improve the performance of the student network using only one modality. And we propose this framework that has been published in MICAI last year and is currently as a journal extension. And then 2 recent works that are actually just submitted. The first one, the question that we ask ourselves is, we might have a lot of images from healthy subjects. In this case, we had 10,000 images from healthy subjects. And the idea was, can we leverage these images and other proxy metadata such as sex, gender, age, to actually build a representation which is then useful for classifying patients, so different kind of patients like Alzheimer's disease or other diseases. And we propose a framework based on contrastive learning to actually learn this kind of representation. And we quantitatively show that this representation is actually useful for classifying several kinds of diseases like schizophrenia, bipolar, or Alzheimer's disease. And what was most importantly, this is very useful if you have subjects coming, if you want to build your representation from subjects coming from one center, so that use a particular kind of MRI scan, a particular protocol, and then you want to use this representation on subjects coming from a different center, so with a different machine and different protocol. And this framework was very useful. It was the results were almost similar to a fully supervised network. And the second, another recent work that we have just submitted to ACML is based on the following idea. Can we actually transfer knowledge or correctly, can we leverage knowledge from a supervised classification to do unsupervised clustering? And here the idea is that we usually have heterogeneous patient populations where we say that is one disease, but it's actually several kinds of disease, so what we call the subtype differences. And so we have the different subtypes of one single disease. And here the question that we asked was, if we want to use unsupervised clustering of this subtype, you should first basically remove what is common, so the variations that are common between healthy subjects and patients, since you only want to focus on the features that are distinctive for the patients. And so here I have just put an example using NIST digits to just grasp you the idea. And on the right, we are just doing unsupervised clustering. So we focus on the digit number 7. And we can see that we have 2 different subtypes, the digit 7 with the trait in the middle and the digit 7 without the trait in the middle. And what we want to do is that we want to discover these 2 different subtypes in an unsupervised way. And we see that if we do a usual unsupervised clustering technique on the right, like k-means, we can see that the 2 groups are actually not found and are not well divided. Instead, if we first do a supervised classification between all the 7 digits and all the other digits, and then we do a non-supervised classification driven by this supervised classification, a non-supervised clustering, only on the 7 digits driven by the supervised classification, we can actually retrieve the 2 subtypes more correctly, and they are highly more interpretable than with the other techniques. And this is, of course, that's just to give you a hint of this idea using digits, but then we also use it on medical images. And we were able to show that these techniques is quantitatively better than other techniques, and it's almost got almost the same result as a supervised technique. And then with a different perspective, we also worked on, can we transfer the knowledge learned on a data set of adults that are usually homogeneous in size, pose, and relative dimension between different organs. Can we transfer a network that has been basically learned on this data set to a completely different data set, so with a completely different distributions, for pediatric images that are more heterogeneous in size, pose, and relative positions and relative shape of different organs. And we propose to do that with geometric transformations. The preliminary work has already been published, and the second basically part of this work is under work. And then the last part, and then I will finish or we wrap up, is about how can we discover anatomical knowledge using artificial intelligence. And here, this is an ongoing research with a postdoc. And here the idea is the following. We have a data set of healthy subjects here on the top, X, and the data set of patients here on the bottom. And what we would like to do is to propose an autoencoder architecture where we first encode what is common between these 2 distributions, between these 2 groups of subjects. And then we also want to disentangle what is common from what is distinctive of these 2 different data sets. And so this will be done with 2 different coders. And then, and that's the most important part of this work, we want to decode these distinctive and common representations in an interpretable way for clinicians, so that we can reconstruct the anatomical traits that were common between these 2 data sets, and then the anatomical traits that were actually distinctive for these 2 data sets. And this is an ongoing research project that will be used with several pathologies. Okay, so that's it. I hope that I've been concise within the 10 minutes. If you have any questions.
Thank you very much. The timing is perfect. So any questions for Pietro? I have a quick one. So you know, you talked about some specific applications and methods applied to those applications, right? And I was wondering how general some of these approaches are. So for example, some of the approaches that you're developing for a pediatric application, can it be generalized so that it can be then, you know, applied more broadly, or does it really depend on the type of images and
some applications were specific, as you say, I can just move on. Like this one was specific for a particular kind of data, which is how can we use adults to pediatric, and this basically tried to work on a specific problems where the question is we have a certain kind of 2 different distributions with basically geometric differences, and how can we go from one to the other? So that's the general question, and then the applications in medical imaging is quite specific, I would say, to adult and pediatric images, but it can be generalized also to others. But then, or I would say that almost all the other works like this one or all the other works are actually quite general and can be applied to any pathology. And so the idea is always like, can we go from basically one distribution of data to the other distribution of data? And then the question is, let's see which are the differences and which are the hypotheses that we can do and try to solve this problem with this hypothesis. But almost all the works that I presented here are quite general and can be used with different pathologies.
But yeah, so I mean, you know, what you were saying about the kind of going from the child to the adult or vice versa could also potentially be applied to something where you might have growth of a tumor or something, you know, you might have like a normal geometry that gets deformed or enlarged due to the, no? Or is that?
Yeah, absolutely. Actually, this work, the one that I presented here, these are actually tumor. And so the idea here is that we are going from a homogeneous population of the adult where the tumor, the size of the tumor and the shape of the tumor is almost all the time is quite similar between each other. And then we want to go to a population where the tumor is actually quite different. Here we have 3 examples of tumor where the shape and the size is very different. And so the problem is that the network has learned on a population where the tumor size and the tumor shape were homogeneous. And if we want to use it on a data set where the tumor size, the tumor shape, the relative shape also between organs are quite different, we need to take that into account. And so the idea here is to do what is usually called domain adaptation. Here we rephrase it as domain normalization. If we want to use it some geometric transformations, so a fine and deformable registration basically done through deep learning networks. And we want to learn that in order to recover, basically, to be able to go from one domain to the other. And it could be adapted to other kind of tumors and to other kind of data, of course.
Thank you very much. Okay, I don't see any other questions, so thank you very much, Pietro.
Thank you.
So the informatics laboratory of École Polytechnique. And he's going to be talking at a different scale altogether compared to the others. So he's going to be telling us about combinatorial rational design and structure modeling of functional RNAs. Jan, all yours.
Yes, thank you very much, Abdul. Thanks for the kind introduction and for the invitation to present our work and maybe more modestly our team. So my name is Jan Ponti, I'm Director de Recherche at CNRS. And I'm currently leading the AMI Bio team at LIX, and that's Abdul introduced. So this is quite, in fact, a change of scale because we are really talking about the nano level here. We are not trying to grasp organs as wholes, but we're really looking at, let's say, the molecular foundations of disease and even in general of biology. So our team consists of myself and Sebastian Will, who's a full professor that's been hired last year as informatics professor at École Polytechnique, and Sarah Berkemer, who just transitioned from a Marie Curie postdoctoral fellow in our team to an assistant professor position at the computer science department of École Polytechnique. We are also greatly helped by Philippe Chassinier and Jean-Marc Steyert, who stays with us as part of an emeritus position. He was the founder of the team and one of the key players of computer science at École Polytechnique for a long time, and also greatly helped by Hélène Thibault, who's our administrative assistant. We have a bunch of PhD students. I'm not going to detail their projects. But we are relatively small teams and a team whose scientific foundation is being renewed with a very strong emphasis on RNA. So one of the only positive outcome of the current pandemics is that I don't have to describe to you what an RNA is, and I don't have to introduce to you the fundamental dogma of molecular biology. So we all know that we have RNAs in our veins and that we can actually supplement them somehow to gain, for instance, immunity to certain disease. And RNA is being increasingly used as a material for biotechnology. So one of these use, of course, is messenger RNA vaccines, which are very promising due to their high level of customizability and lack of let's say, adverse effect, at least so far. But also, RNA, it's probably less known that RNA is at the core of new genome editing techniques, like, for instance, CRISPR. That's probably the future of gene therapies, so to speak. And CRISPR is usually a complex made of proteins, but the targeting mechanism within CRISPR is actually performed by RNA, and a very rational design of RNA is really beneficial to avoid adverse effects of these technologies. RNA can also in itself sense the presence of metabolites and then reveal concentration of those metabolites by undergoing conformational changes upon binding with these small ligands and expressing maybe reporter proteins down the road. That's what is called the rubber switches, and it's also the object of excitement in the biotechnology community. But also RNA and the concentration of different transcripts within the cell reveal many things about disease and are very helpful for diagnosis and prognosis, especially in the context of cancer. There's also a lot of involvement of RNAs in genetic disease that are not associated with coding regions outside of proteins. And in that context, some mutations can be associated with either changes of the structures adopted by regulatory regions, or even have a much more subtle effect by changing the equilibration between different type of conformations in solution. And RNA is also at the core of some of the viruses that are the pathogens that are the main trouble we are facing right now. For instance, the genomic material of HIV is composed of single-stranded RNA, for which knowing the structure is actually important to understand how to fight against HIV. But it's also the case for SARS-CoV-2, the pathogenic agent of COVID. Also, back to the therapeutic level, there's been quite some work on silencing RNAs and RNA interference techniques. And there are currently, I think, 3 FDA-approved drugs that are based on this technology for usually rare genetic diseases. So RNA is really ubiquitous. It's very versatile. Actually, if you look at the RFAM database, which kind of clusters RNAs depending on their function, more or less, one can find as much as 3,000, little over 3,000 now, functional families of RNAs, which have usually homologous across organisms, across organisms, and perform a variety of functions. But from a computational perspective, all those biotechnology applications essentially narrow down to 2 types of problems. The first one is modeling the structure of RNA. And usually we do that at a secondary structural level, meaning that we don't want to know precisely where atoms are in 3D, but we just want to know which nucleotide binds to whom. And this representation is usually enough to already formulate a functional hypothesis. And once you unlock this capacity to predict or to understand how RNA folds, then you can start asking questions regarding RNA design. So you want to essentially formulate RNAs that fold in a certain way or encode a certain protein and generally perform a certain function. So I'm going to first talk a little bit about the folding process. So you start from a sequence, for instance, and then you have some base pairing rules. Let's say A with base pair with U, G and Cs, and G and U, and you have an underlying free energy model. And then you have a wealth of different structures that can be adopted by your RNA. In general, it's an exponentially growing family of such conformations. And finding, for instance, the conformation having minimal free energy, so the most stable one and the most likely one to be observed at the thermodynamic equilibrium, is really a difficult task due to the sheer number of structures that can be adopted. So even for this modest size transfer RNA, for instance, you can see that there are many different structures. And so instead of brute forcing our way through all of the possible conformations, or maybe finding some kind of gradient descent strategy, what we can do is use an algorithmic technique called dynamic programming that essentially decomposes the search space in a clever manner to avoid recomputing things that have been already computed. And that yields n-cube algorithm, so polynomial time algorithm, polynomial on the size of your input RNA, in order to, for instance, produce the minimum free energy structure out of this huge diversity of possible conformations. So you can supplement the energy model with experimental evidence that are usually produced in a matter of weeks in the labs. So I'm talking about chemical probing techniques or enzymatic probing. And you can also combine these algorithmic techniques with the knowledge that multiple homologs of an RNA across organisms will fold in a certain way. And then you can perform a simultaneous folding and alignment of RNA, which is at the core of most of the successful comparative prediction methods. You can also sample a bunch of representative structures within the Boltzmann-Gibbs ensemble using exact sampling as opposed to Monte Carlo Markov chain. And that unlocks many different applications and many different methodological tools that you can use. So one example is the IPANEMAP method, which essentially predicts structures that are equally supported by different type of probing protocols. And so towards that, we perform this kind of sampling coupled with unsupervised machine learning, so clustering, in order to get structures that are equally supported by several reagents. That's just an example. And we can apply this kind of method in order to understand better, for instance, the biology of viruses. So here we worked with the Sargay lab in Paris Descartes in order to come up with more precise structural models of the HIV gag virus. And these experiments coupled with other with toehold experiments allowed us to discover new recruitment sites for the host ribosome by the HIV. We also have had an ongoing collaboration with Chinese scientists, and we used similar algorithmic techniques applied to RNA-RNA interaction prediction in order to discover an alternative template switching mechanism in the replication of the genetic material for SARS-CoV-2. And once you understand how to fold RNA, then you can start asking, how do we design new RNAs? And unfortunately, the problem is much harder. So here I just present to you just one type of design scenario, but there are really many different types. You may want to design, for instance, an RNA which will undergo conformational change upon binding of a ligand. At that point, you're designing sequences that are supposed to adopt several different structures potentially, and have some energetic consideration on top of that. And unfortunately, the algorithmic problem become intrinsically much harder. So they are sometimes NP hard, or they are sharp-P hard, which is the counting equivalent. And those are complexity classes in computer science that show how hard it is to solve a problem. And we used a category of techniques that are used parameterized complexity, where your algorithm is exponential, but only on a parameter that's not the size, but something much smaller in practice, and to solve the problem not only in theory, but also in practice. And we contributed tools towards that. We also showed that those techniques could be adapted to do negative design, which is essentially ensure that your RNA folds in the right way. And more generally, we are developing and we are really looking for avenues to really benchmark our techniques to perform an efficient and flexible design of new RNAs under a very rich set of constraints that arise from a specific application domain. So just to summarize a little bit who we are, because this was more a talk about introducing the team rather than trying to really talk about science very deeply. We are a team of computer scientists that have really deep foundations on the theoretical computer science side and discrete mathematics. Our tools of choice are essentially constraint satisfaction and also really deep focus on algebraic dynamic programming, which is generalization of the techniques I talked to you about. We use and we can maybe even sometimes contribute a little bit to machine learning, but we try to avoid it as much as we can. because we prefer mechanistic models, and we are at a scale where we can afford to talk about physics, so we don't necessarily need to use black boxes. Okay, but and I'm open to questions and controversies regarding that. But we use it from time to time, because sometimes statistics is the best way to approach a problem. And we try to develop as much as possible exact methods under the assumptions of models, of course, which can be questioned, for structural modeling, rational design, so prediction of interactions, and also we have methods for visualizations that are quite popular in the community. And we try to apply not only as a way to showcase our tools, but also as a way to inspire future developments. And some of our applications domains are on RNA viruses, especially the molecular biology, cancer transcriptomics, but that was mostly in collaboration with Paris Saclay. And right now we have ongoing projects on design of aptamers against Alzheimer's. So RNA molecules that would bind to certain proteins associated with Alzheimer's disease in order to disrupt the disease, more or less. And more generally, we have more abstract and more theoretical interest in RNA evolution, but that informs future developments in our work. And with that, I'll just stop and maybe take a few questions if time allows.
Thank you very much, Jan. Are there any questions? Again, you can ask your questions either through the question answer, the chat, or through the or by raising your hand. Okay. I don't see any questions. I think maybe we will stop right here, Jan. Thank you very much for the presentation. And thank you for all the speakers for staying on time, and that allows us to not fall further behind because of my problems at the beginning. So now is a break time. Normally the break was scheduled to end at 11:10, which is now. So we're starting the break at 11:10. If you don't mind, just to kind of reduce our delay, I'd like to cut the break down to 10 minutes instead of 15. So if we can start again at 11:20, that would be fantastic. Thank you very much. And the next session is going to be chaired by Anne-Chantal Gouget, and it's going to be the focus on some of our young researchers. Thank you. See you in a few.
Thank you.
bonjour.
Bonjour.
Notre première speaker.
Oui. Les personnes vous entendent. Hein. Vous êtes en direct. OK.
OK, on n'a rien de grave, mais... mais merci de nous prévenir. Et donc, les étudiants sont en train de se connecter.
Salut Chantal.
Bonjour Abdoul.
Ça va?
Oui, ça va, merci. Bon courage pour toi.
Merci. Merci beaucoup pour ta participation aujourd'hui.
Mais je t'en prie, c'est un plaisir d'être là.
Je crois qu'on peut répondre.
C'est l'heure, on peut commencer. So I switch in English.
Yes.
Okay, thank you. So good morning, everybody. My name is Anne-Chantal Gouget. I am a research director at CNRS, working in the condensed matter physics laboratory at the École Polytechnique. And I have the pleasure to be the chair of this session, which is dedicated to our young scientists. So it's a dense session with 10 presentations. And therefore, I will really ask to our speakers to respect the scheduled time of 7 minutes. And if you agree, I will inform you at the end of the talk. Then we will have 3 minutes for the questions. And for this, dear audience, I really rely on you to ask many questions to our young students because it's very important for them. And for this, please only raise the hand, no chat, please, and I will call you. I think it will be more dynamic for the discussions. And of course, if there are too many questions, do not hesitate to use later on the chat to directly ask your question to the speakers. Okay, so now we can start with the first talk, which is given by Cristina Cardenal Peralta from the Biochemistry Lab at the École Polytechnique, and which is about modulating eIF2 alpha activity for cardiomyocyte rescue. Please, you can start.
Thank you so much for having me and giving me the opportunity to present my research. So in the last decade, endoplasmic reticulum stress has emerged as an important mechanism in cardiac disease. Upon cardiac injury, the ER responds by activating the ER stress response. When this response is mild or short, cardiomyocytes are able to restore cellular homeostasis and survive. However, when this response is prolonged too long in time, cardiomyocytes go through apoptosis and are replaced by scar tissue. This response is controlled by the activation of eIF2 alpha. Upon phosphorylation of this protein, this cascade occurs. So this gives us a lot of space to study the control of cardiomyocyte fate by modulating eIF2 alpha activity. So that's exactly what our collaborators in the interim lab of Christophe Lemaire were doing. And they were studying this pathway when they discovered that eIF2 alpha is not only phosphorylated, but it's also acetylated in a lysine. Moreover, they discovered that when it's deacetylated, cardiomyocytes are able to be rescued from this apoptotic state and restore cellular homeostasis. This is controlled by a protein called SIRT1. Now, what is even more interesting about SIRT1 is that it can be specifically modified, its activity can be specifically modified by sirtuin activating compounds, also known as STACs. So this became the basis for my PhD hypothesis, which is that SIRT1 activity protects cardiomyocytes from apoptosis. And my two main aims became to understand the mechanism by which SIRT1 deacetylates eIF2 alpha and to modulate SIRT1 activity by discovering specific STACs that modify its activity. So in order to do that, I need to produce both proteins. And in order to study this interaction, I need to decipher their protein structure by using crystallography assays. And in order to study this, or how this STACs, this compounds modify SIRT1, I have developed some enzymatic tests that allow us to do so. So this research entails two main problems. The first one is that all previous measurements done with SIRT1 have been done using mimic peptides. So there's no real information on how the enzyme acts with a real substrate. And the second thing is that this substrate that we want to use, EIF2 alpha, and its acetylated version have never been produced before in E. coli systems, which are the systems that we use in the lab to mass produce proteins. However, there's always a solution, and we are able to engineer acetylated EIF2 alpha. The main problem is that acetyl-lysine is not an amino acid that's present in the genetic code. However, using an orthogonal system developed a few years ago, we're able to expand the genetic code. And this is based in stop codons. We all know that one codon codifies for one amino acid, and that these codons are recognized by tRNAs, all but stop codons. So what we can do is reassign one of these stop codons to this unnatural amino acid, acetyl-lysine. So here you can see the sequence, or like a picture of like the sequence of our protein with the codon for lysine. So what we do is modify that codon for a stop codon that will reassign to an acetyl-lysine. In order to do that, we need two more things. We need an engineered tRNA that's able to recognize this stop codon. And we also need an amino acid tRNA synthetase that can put these two together. So what will happen is we'll have our tRNA, our acetyl-lysine amino acid, and this newly engineered synthetase that can put them together, and then can be recognized by the ribosome and then be introduced in our protein and be able to express it. However, EIF2 alpha wild type by itself without any modification, it's already very difficult to produce using E. coli systems. So we optimized EIF2 alpha sequence so it could be better recognized by the E. coli bacteria. And we also used a chaperone system that allow us to express it better. So after a few months of optimization and hard work, we were able to produce both. So what you can see in this gel in the red box is EIF2 alpha non-acetylated, the wild type protein. And what you can see in the blue box is the acetylated protein. We need to verify that the protein is indeed acetylated in the lysine that we want to. And to do that, we sent the sample to mass spectrometry, and we were able to confirm that our protein is indeed acetylated. Then we need to produce SIRT1. SIRT1 does not present any problem when expressed in E. coli systems, so that was very easily purified compared to EIF2 alpha. What is very important about SIRT1 is that it has an N-terminal domain, which in the picture you can see as the purple domain. That is a domain to which this stack compounds will bind. So it's very important to produce a protein that has this N-terminal domain. So now that I'm able to produce both proteins, I was able to do some enzymatic tests. So first, I did some tests with SIRT1 and a mimic peptide, and SIRT1 and the acetylated EIF2 alpha. And as you can see in this picture, this results strongly suggest that SIRT1...
1 minute left.
Okay, SIRT1 has 4-fold more affinity than for its mimic. So this means that using the real substrate gives us more chances of discovering specific compounds that will then modify its activity according to this substrate. And now that I'm able to produce both, I can continue to characterize the interaction between these two proteins. And I will start to try and find specific stack compounds using the SciUpload platform at the Châtenay-Malabry faculty, where I will be able to test more than 3,000 compounds. And finally, I will continue to analyze the interplay of phosphorylation and acetylation together in EIF2 alpha and how it affects this whole pathway. And finally, I'll just like to thank the École Polytechnique for its funding, my colleagues in Bioc for the help and support, our INSERM collaborators. Without them, this research will not be possible. And finally, the SciUpload platform that will allow me to do this high throughput screening. And finally, thank you for your attention.
Okay, thank you very much for this very nice presentation. So we have time for questions. So I don't see no hands.
That's all right.
So, Sylvia, yeah, no.
Yes. No, I don't see any questions.
Okay, so we thank the speaker again, and so we will move to the second talk, which is given by John James from also from the biochemistry lab, and which is about vinsulin regulates collective cell behavior by remodeling the actin cytoskeleton at cell-cell junctions. Please, you can start.
Okay, I hope you can hear me now. And can you see my screen?
Yes.
Yeah.
Okay, so I'm going to be talking about my project, which is vinculin regulates collective cell behavior by remodeling the actin cytoskeleton at cell junctions. Yes, so in a normal human body, tissue development requires cells to collectively regulate their migration, their cell junctions, as well as their proliferation. And any failure to regulate these processes can lead to diseases like cancer. Cells regulate them by integrating biomechanical as well as biochemical stimuli from the environment to modulate their actin cytoskeleton. For this project, we focus on mechanical stimuli, which are transmitted to the cells through cell matrix and cell-cell adhesions. Here you can see a structure of these adhesions, and you can see in both cases that when there's a tension applied on these structures, vinculin is recruited. Interestingly, vinculin binds to R2-3, which is the protein that the cell requires to create branched actin networks, which are important for all three of these processes. So the goal of my project is to understand how vinculin regulates cell collective behaviors, and I will be looking specifically at the role of R2-3 at cell junctions, as well as the cell density dependent control of proliferation and migration. So to answer these questions, I first created a vinculin knockout cell line using CRISPR in an MCF10A cells, which are non-tumorigenic mammary cell lines. You can see that there is no vinculin in cell junctions here. As a result of this, while parental MCF10A cell lines prefer to stay together and migrate collectively in a 3D matrix, vinculin knockouts are much more independent. We quantified the rate of cell junction disassembly, and you can see that vinculin knockouts have a lower cell junction stability. We think this is because there is a lack of R2-3 recruitment to cell junctions, to early cell junctions in the vinculin knockout. To confirm this, we transfected MCF10A cells with a small part of vinculin that binds to R2-3 and to pull down R2-3 along with this peptide. So here that the kinetics of interaction of R2-3 to the peptide recapitulate the kinetics that we see here in recruitment. Now, to see the effect of this on collective behavior, we first looked at how cells proliferate. We created growth curves by just counting the number of cells in a field at a certain point of time, and we see that vinculin knockouts proliferate much faster and they reach a higher saturation density compared to parental MCF10A cell lines. We confirmed this using an EDU assay, where we look at cell cycle progression, which has been shown to be dependent on R2-3 activity. We saw that the vinculin knockouts keep proliferating even at a higher cell density compared to parental MCF10A cells. This suggested that removing vinculin increases R2-3 activity. To confirm this, we looked at phenotypes that we know are dependent on R2-3, the first being cell spreading, because R2-3 creates the branched actin which pushes the cell membrane outward. So that vinculin knockouts indeed have a higher area than parental MCF10A cell lines. We then looked at single cell migration as well. You can see tracks of cells migrating randomly, and the vinculin knockouts have these cells which move very persistently in one direction. We quantified this by calculating the autocorrelation of angle of migration of these cells. This This has been shown to increase with R2-3 activity. Here again, we see that the vinculin knockouts are much more persistent, and all of this shows that vinculin antagonizes R2-3. Now to look at collective migration, we allow cells to grow to saturation in a monolayer, where they stop dividing and they stop moving. This is called jamming. When we then treat them with a hypotonic solution, we see that they suddenly start moving again. And this movement is constrained within domains. We analyze these pictures using particle image velocimetry, where we get these nice displacement vectors, maps, from which we can derive vorticity maps. And you can see here that the distance between boundaries of vorticity is much higher in parental MCF10A cells compared to vinculin knockouts, indicating that they may have larger domains and therefore are more collective. We quantified a correlation distance, which indeed shows that vinculin is required for coordination of cell migration. To look at the molecular reason behind this, these cells for actin. And we see in our parental MCF10 lines that there are these long fibers of actin that go across cells and they are coordinated over a large distance. While in the knockouts, we have small fibers which are at most connecting cells. And of course, such a huge difference would have an effect on tissue physiology. So we looked at acini, which are the closest you can get to a mammary gland in cell culture. You can see that the normal acini is spherical. It has a lumen, and the cells secrete laminin to create a basal membrane. Our vinculin knockout cells form acini that are not spherical, they do not have a lumen, and there are defects in laminin secretion. This indicates that vinculin is essential for morphogenesis as well as maintaining tissue integrity. So I would like to conclude with a working model that we have shown that vinculin recruits R2-3 to cell junctions. Since vinculin antagonizes branched actin formation, it leads to formation of more linear networks, which can then be bundled into these long fibers and coordinate cell migration. I would like to end by thanking mainly my supervisors, Stefan and Alexis, of course, everyone at the lab who I learned a lot from. And over the course of the project, we had discussions with a couple of other professors, Pascal Servazan and Christophe Leclerc. Thank you for your time.
Okay, thank you very much for this presentation. So we can ask questions for John. So no questions again.
I have a question actually, sorry.
So okay.
So vinculin affect only caderons or also other families of cell junctions molecules?
So far, we know that it affects adherent junctions as well as focal adhesions. And when a tension is applied, you know that alpha-catenin is stretched, and stretching of alpha-catenin reveals a vinculin binding site. So vinculin is recruited. So it's to adherent junctions.
Thank you.
Okay, so do we have any other question? No? Okay, so we can thanks again our speaker for this nice presentation. And then I will move to the next one, so which is given by Olga Markova from the LADIX at the École Polytechnique on mechanical probing of tumor infiltrating lymphocytes. Please, you can start.
Hello, everybody. I'm Olga. Can you see my screen?
Yes. No, no, we just see you.
Thank you, organizers, for giving me opportunity to presentation.
Excuse me, we don't see your presentation.
You don't see presentation?
Yeah, okay. Yeah, it's perfect now.
Thank you. Good. And so thank you all for finding time to connect in for my talk. I will present mechanics of tumor infiltrating lymphocytes, the project I work with Juliana Son at École Polytechnique and Claire Rose and Emanuela Romana at the Institute Curie. Programmed cell death protein 1, also known as PD1, was discovered by Tasuki Honda in early 90s as a molecule whose disactivation increased immune system response. This discovery raises a huge hope to beat cancer in the very recent future. So what makes PD1 so valuable for immunotherapies? Lymphocyte is moving in a lymph node and by chance meet antigen-presenting cell, APC. It recognizes via receptors. The APC recognition leads to changes inside the lymphocyte, significant cytoskeletal reorganization. At the cell level, lymphocyte flatten contact with the APC, and then it makes protrusions that is very active mechanical quantities that make a lot of pushing and pulling forces. Interestingly, during very first minutes of lymphocyte contact, the stiffness of lymphocyte increase and jump up several times. Stiffening of lymphocyte increases its resistance to eventual mechanical shocks that it can encounter while he's in his crowded lymph node. Furthermore, stiffening reflects the actin cytoskeletal reorganization, which is indispensable step in the establishment of immune response and remove antigen-presenting cells, including cancer cells. Then to not overstimulate immune system, lymphocyte can also express PD-1 receptor. If PD-1 recognize PD-1 ligand, PDL1, on the surface of antigen-presenting cells, lymphocyte is not activated, which is okay in healthy case. However, in the case of cancer, this decrease lymphocyte activity and prevent immune system from killing cancer cells. Immunotherapy drugs that block PD-1 engagement re-establish the immune defense. They are very promising tool for cancer treatment. However, fundamentally, we still know very little about how PD-1 action inside the cell, what it's doing, especially how mechanical things are changing in PD-1 engagement. And here we address this question. What the effect of PD-1 engagement and the mechanics of lymphocytes? This project contains several steps, set the model, mechanical measurement, PD-1 engagement, work with tumor infiltrating lymphocytes, those that extracted from the patient tumors. I started my postdoc just 6 months ago, and today I will present you the first part of the project. The model. We use Jurkat cell to mimic lymphocyte, and for the APC model, we use beads that recover these antibodies. We then put together beads and the cell and observe the contact between them. Here you see the cell that meets the bead on the top and immediately start to make protrusions. Another cell have two beads to take care about, and it makes two protrusions. This is how we know that lymphocyte is get activated. Now our goal is to measure mechanics, and for that we use micropet force measurement. We hold cell by glass stiff pipette and apply little negative pressure, and the bead is brought with flexible micropipette with known stiffness. Using piezo microstation, we move a bit to the cell resists and flexible bead bands. So here we use image analysis to calculate deflection of the pipette, and from that we can now calculate the force that exerted by the cell, and we can do it with pico-Newton precision, which is very high. It's like single molecule measurement almost. Now, here you see the example of experiment of micropipette force measurement experiment, where leukocyte is searching the bead, and here is a quantification of stiffness that increase up to 2.5 times just in 3 minutes after the contact. actin is also accumulated at the bead contact. So we can see it by transfecting lymphocyte with actin. Now, to make our model even more close to a biological case, Naomi, a PhD student, created an APC-based cell model where control cells is coated with antibodies and PD-1L cells express PD-1L ligand. Jurkat said put in contact with artificial APC, make numerous protrusion and explore APC cells. The front of a lymphocyte changes shape, then lymphocyte also retract. You can see at the end of the movie from the pipette, and this indicates that the stiffness and possibly tension of lymphocyte increase. Furthermore, actin strongly accumulates at the protrusions at the cell-cell control, at the control.
1 minute left.
Comparison with APC cells expressing PD-L1 shows that actin does not accumulate at PD-1 expression cells as efficient as in control situation. So this clearly indicates impairment in the establishment of immunological synapse in the case of PD-1 engagement. We finally adapted micropipette force measurement setup geometry to be able to measure lymphocyte stiffness while it interacts with APC. With that, I would like to conclude that we found that PD-1 engagement perturbs actin accumulation at immunological synapse. Jurkat stiffness increases by contact with activator bead, and we have a setup to measure mechanics in the case where cell-cell model we used. Our next step would be to measure effect of PD-L1 on engagement of Jurkat and work with tumor-inflating lymphocytes from patients. I would like to thank Julien Husson and Claire Ross and Emanuela Roman to set up the project and for very valuable help. Noyem and Stephanie for all biological part of the project, Abdul Barakat for hosting me in the LADIX, and of course Foundation Belancourt to make it all possible. We'll be happy to hear from you.
Okay, thank you very much for this very nice presentation. So are there questions? I see no hands. Otherwise, I have one. Just by curiosity, how do you functionalize your microbeads with your antibodies and your PD-L1 cells, please?
So to functionalize with antibodies, we incubate them with them simply. With some we have a protocol that works fine. And for the cells, we code them with antibodies, and for PD-L1, it's expressed, it was created by Naomi genetical cell line, so she did it constantly expression of this protein on the surface of the cell.
So it's much more an adsorption process on the microbeads?
So, excuse me?
It's an adsorption process. It's physisorption of your proteins on the microbeads.
So the beads is easier to do than cells, if this is the question.
My question was, is you put some sort of covalent attachment, for instance, between
the streptavidin beads
coated, or
yes,
it's just
already coated by streptavidin, and then we have anti-CD3, excuse me, anti-CD28 antibodies that we incubate together, and that's how they are attached to the bead. Yes, of course.
Okay. Okay.
We need to.
Thank you very much.
Excuse me for.
I think
there are a couple of questions,
but actually I don't see them, Sylvia. So
if you go, if you have to click on participants, only you can see. So the first question
I see only my hands actually.
Oh, okay.
So I don't know what's okay.
If you want, I can.
Yes, please. Mm-hmm.
The first question is from Sliman. Please, you can talk. Sliman, you should activate. Okay, no.
So I see on the chat also Emmanuel Bourpierre has a question.
Okay.
Yes, Emmanuel, you should activate your micro.
Oh, can you hear me now?
Yes.
Yes.
Okay. Yeah, the experiment with the bead and cell is very nice and fun. And how hard is it to achieve it? I mean, does it work every time? Or do you have to do many bead cell experiments to be able to observe it? And is there a way to automate it? Or, I mean, how does it work?
It would be great to make it automation. I thought about, we have like 5 pipes that connect 5 cells. Well, we are not here yet. We do it manually, cell by cell. We have some rate of response that depends on different parameters. So, well, cells need to be in good health. We see that not all of them respond. On the glass experiments, we have like 70% of responding to the activation, and so we take into analysis those that respond. So this is how we deal with that.
That's nice.
Okay, Sylvia?
Yes.
Other questions?
No.
Okay, so we thank you very much again, Olga, for this talk. And now we will listen the presentation of Fanny Mousseau from the lab at the École Polytechnique on the Europium DOPS nanoparticle-based multiplex lateral flow immunoassay for the quantitative and highly sensitive detection of enterotoxin. Please. Okay, we see your presentation.
Thank you for the introduction. Good morning, everybody. So I'm Fanny Mousseau. I'm working at the lab LOB with Antigone Alexandrou and Cédric Bouzig. And I'm developing a portable, rapid, and ultra-sensitive in vitro diagnostic test based on luminescent nanoparticles. So if you want to develop a fast, cheap, and portable test, it is recommended to work with lateral flow assay. And you know this kind of test because pregnancy tests sold in pharmacies and antigenic tests developed for the detection of COVID are LFA. And how it works, so basically you have your sample with the analyte and you add nanoparticles coupled to antibodies, and the antibodies recognize your analyte. And then you dip a strip in the sample, and on this strip you have 2 lines, a test line and a control line. And then the sample migrates along the strip by capillarity. And if the analyte is present, it will be sandwiched in between antibodies on the test line and antibodies grafted to the particle. And since you are able to see the particle, you can see if the analyte. You can detect the analyte. So in commercial assay, the nanoparticles are gold nanoparticles, and in absence, and so you obtain this kind of strips. So in absence of analyte, you see only 1 red band, which is the control line. And in presence of analyte, you have 2 lines, the test line and the control line. And the problem with this kind of assays is that they are not sensitive and not quantitative. So to circumvent these drawbacks, we replace the the gold nanoparticles by luminescent nanoparticles. So these luminescent nanoparticles were developed by Thierry Gapin is working in the lab PMC at Polytechnique. And so we carried out the strip migration as explained previously. And then to visualize the test line and the control line, we use a homemade portable reader. So basically we place the strip in the reader and we illuminate the strip in the UV. And then we take strip images with the camera of a smartphone. And we developed a smartphone application, which is able to control the image capture, and also which can run the image analysis. So briefly, what does the application? So we plot the profile of the test line along the direction of the migration. And then we defined our signal as the ratio between the amplitude of the luminescence over the background. Sorry. And so our goal is to build calibration curves. And finally, if we want to study a sample with an unknown concentration of the toxin, we just run all this cycle of analysis. And from the measurement of the signal and from the calibration curve, the application gives you the analyte concentration. So I'm working with toxins that are called staphylococcal enterotoxins. They are responsible for food poisoning and they may be used as biological weapons. So it's very important to be able to detect them on field with a portable, fast and sensitive detection. And here I will show you my results for 4 different toxins, SEA, J, H, and I. So basically, I performed assay with gold nanoparticles and luminescent nanoparticles. And each time you see 2 lines, the control line and the test line. And you see that the test line, the intensity of the test line increases when the toxin concentration increases. And with the luminescent nanoparticles, we are able to detect lower toxin concentrations compared to the reference assay. So I did these experiments for the 4 toxins, and just by a visual inspection of the strips, I observed that with the luminescent nanoparticles, we have gain sensitivity ranging between 5 and 10. Then I performed the quantitative analysis of the strips, and I was able to accurately fit the data, which means that we can obtain a calibration curves for the different toxins, which means that we are able to perform quantification of unknown samples. With more details, so when we do the quantification, we have gain sensitivity between 2 and 4, meaning that thanks to the luminescent nanoparticles and the quantification, we have global gains in sensitivity between 15 and 40. So with our system, we are able to do a detection which is quantitative and sensitive. Then I did a multiplexed assay, which means that I detected several toxins, so in this case 3 toxins, at the same time in the same sample with a single strip. And I performed again the quantification. And by using the calibration curves obtained from the simplex experiments, I was able to retrieve the nominal concentration of the toxin, but to a factor.
1 minute left, please.
Thanks. To a factor of between 1 and 3, which means that the detection is only semi-quantitative. But we obtain similar sensitivity as for the simplex assay. And it is an important point because usually when you multiplex, you lose sensitivity. And so finally, we obtain sensitivity which are higher than in the commercial ELISA, and that approach the sensitivity that we obtained with our optimized ELISA performed with the same set of antibodies. So finally, with our system, we implemented a portable, rapid, and ultra-sensitive detection. And to finish, I would like to thank all my collaborators, and I thank you for your attention.
Okay, thank you very much for this very nice talk. So I see that Abdul Barakat has raised his hand, so please, you can ask your question.
Thank you. So my question is, for the multiplex assay, do you use the same calibration curves that you use for the single analyte assay, or do you do specific calibration curves in multiplex?
No, for the moment, we use the calibration curves made for only 1 toxin, but it doesn't work, but it's not so surprising. And so at the moment, I'm trying with an intern, which is called Melissa. So we want to do several 100 different triplets of concentration to have a map and to try to have a 3D calibration curve.
Yeah, thank you, Annie.
So I don't know if there are other questions. Otherwise, I have one.
Mm-hmm.
Are there questions or not? No, I think so. In your case, you use this sandwich method to detect the analytes. So my question is the following: are there commercially available antibodies dedicated to the toxin you used, or you have to produce them, or how do you proceed?
So we can buy them. For instance, we can buy the antibodies available for the ELISA assay.
Okay.
But they are not very good. So we have a collaboration with the CEA, and they produce antibodies. And they have really nice ELISA results with their antibodies. And I think with their antibodies, they have maybe they are 100 more sensitive than the commercial assay or something like this.
Okay. Okay.
And that's why we are very proud to be quite close to the ELISA, the sensitivity obtained with the optimized ELISA, because it's a good sensitivity.
Okay, thank you. So thank you again. And so now we will continue with Ouda Assini from Samovar at Télécom Sud Paris. And so the talk is about, so it's in French. Oh yeah, okay. Thank you for the translation. On complex value deep neural network for augmented microscopy applied to malaria diagnosis.
Can you hear me?
Yes.
Okay, so I'll share the screen again.
We don't see you.
Okay, sorry. Okay, is it okay now?
Yeah, we see you, but we lost your presentation.
Okay.
Otherwise, it doesn't matter if we don't see you. Okay, it's perfect now.
Okay, perfect. So I will start. So thank you for the presentation. So I'm a first year student, PhD student at Télécom Sud Paris, and I will be talking about complex valued neural network for augmented microscopy applied to malaria diagnosis. So as we all know, malaria is a dangerous disease that is still causing a lot of death, even if it's curable. And this is why the World Health Organization is recommending testing as a strategy to stop this virus. And what they require is, what they recommend is a microscopy test that is based on observing blood smears under the microscope and detecting cells, red blood cells, and then checking if they have any parasites in them or not. And basically, to make your diagnosis reliable, you have to check over 200,000 cells. Otherwise, you cannot conclude. And this is why it's hard to apply this testing method in real life cases. For pandemic zones, for example, and it was the born of automated analyzer for the malaria diagnosis, which are basically techniques based on capturing some images from microscopy and then analyzing them and segmenting or classifying them into malaria or not malaria using deep learning approaches. But until now, the majority of these approaches have been low sensitive or maybe too complicated to be used. So what we propose today, what we suggest today is the introduction of a new information like which is phase information. In fact, phase information is really detail relevant, even if you have seen truth objects. So for example, here you have a seen truth frog membrane, so you cannot detect it in here in intensity classical imaging. And when you use the phase contrast imaging, you can see that you can detect the form and maybe the particularities and the details of this membrane. And we were asking ourselves what will be the impact of using such an information in addition to intensity information in the detection and the classification of malaria, and what it is the best way to introduce this information, because it's like complex valued information which is not adapted for classical neural networks. So I will start with the acquisition of the data set. What we use is a biophotonic device developed by Telecom Sud Paris. It's inspired by high-resolution Fourier picographic microscopy, which is a way the way of imaging that consists of eliminating with different angles, and then given some correction in the spectrum, you will have the intensity and the phase image. I will give more detail maybe if it's not clear at the end. So what I wanted to show here is that comparing to classical microscopy at magnification 100, you can, using our technique at magnification 20 only, you can see that we have a lot of details. For example, hemozoin that appears clearly here, and we cannot see them clearly in the first image. But also the phase information is a kind of plus, because you can see the vacuole here and the nucleus, which may be a better way for characterizing the malaria parasite. So we can see it clearly, what we could not do at this stage with this microscopy system, which is classical system. So after getting the data set, which is composed of intensity and phase images each time for each cell, we try to classify those images into parasite, non-parasite images. So we choose to use a neural network called faster RCNN, which is basically a combination of two neural networks. The first one here is the one responsible of proposing, of suggesting some cells, some regions of interest, basically the red blood cells. And then the second part here will allow us to classify them into parasite or not parasite. And they are sharing here an extractor, which is basically a way to extract the most relevant information in your input image and then give it to the two sections in here. As you can see, this kind of neural network is designed for maybe classical images with RGB channels, and we cannot put complex imaging in here because it will destroy the phase information. It's not adapted. So what we suggest is to change this part here and made it like a complex valued filter to extract the same information, but with another type of filter more adapted to the physical problem of bimodal information, phase and intensity. We changed the type of the input. So we don't give phase and intensity, we gave the real part and the imaginary part of the image. And then we transformed also the filters. So they are complex filters now, and they were real filters only at the beginning. And we performed classification using this new network. So we performed some tests on a quite high number of red blood cells, but only coming from 2 patients. And we have a more classical parasitemia proportion. So what we investigated is the impact of introducing phase and the impact of using convolutional, complex valued convolutional networks The first thing that we have seen is the impact on the number of trainable parameters. So you can see here that we have a lower number of parameters for a complex valued neural network. So you're only learning the half of the parameter in other cases. And this model will be much less complicated and will be simpler maybe to use in daily life and to give faster predictions. And as we can see, we can see that introducing phase information have enhanced the sensitivity of the system. But the most important enhancement is only observed when we use complex convolutional value to extract the information from the image. So our go-to method will be the complex valued neural network with the introduction of phase information. So that's it for the results. So to sum up, I will say that we have introduced a kind of imaging that already exists, but were not explored in case of malaria, and that allows us to detect maybe more morphological properties and details. This information gave us a better result on sensitivity. But only through a complex convolutional neural network. And that's it for the result. In our further work, we will like to perform...
I'm sorry, you have to finish. I'm sorry.
Okay, no problem. So we will just perform other experiments, and that's it. So thank you for listening.
Yeah, I'm sorry, it's my fault. I missed the time.
No problem.
We have time for one or two questions, if there are. I don't see questions. Okay, so thank you very much for this nice talk. Now we will listen Lena Chanchilia Atencia from Biomaps at University Paris-Saclay, and is going to talk about kidney perfusion mapping by ultra-fast ultrasound imaging for non-invasive diagnosis of chronic nephropathy.
Hello everyone. Can you hear me?
We can hear you.
And can you see my slides?
Yeah, exactly. Everything is perfect. You can
now you can see when I pass the slides?
Yes.
Perfect. Okay, so thank you very much for the invitation. My name is Lenin Chinchilla, and today I will present you part of the work developed in the perfusion project. This project is carried out in collaboration with Dr. Jean-Louis Janison and Professor Jean-Michel Correa, and financially supported by the École Polytechnique and Foundation Bettencourt-Schweller. In a clinical context, the chronic allograft nephropathy is a pathology that affects the transplanted kidney and is characterized by the formation of fibrosis. This pathology can lead to graft loss and is highly prevalent. The methods currently used to monitor the transplanted kidney are based on biopsies, ultrasound Doppler, and MRI. However, these methods don't allow early diagnosis of CAN. So the objective of this project is to develop a non-invasive technique as a new diagnostic tool to understand and characterize the processes underlying CAN. How to do that? So the idea is to combine two quantitative ultrasound techniques, elastography and ultrafast Doppler, to get a resistivity index map and information about internal pressure. This could be of great importance for the diagnosis of chronic allograft nephropathy. In brief, these ultrasound sequences are performed as follows. In elastography, a focused ultrasound beam is used to generate a radiation force at different locations in the medium, followed by ultrafast plane wave imaging at a very high frame rate during 40 milliseconds. This provides a propagation film like this, from which we can estimate the shear wave speed that is related with the shear modulus. In ultrafast Doppler, the same ultrafast plane wave imaging approach is used to map the flow blood. Then these acquisitions are filtered to remove the clutter signals and to get signals from blood. from which we can compute the power Doppler image and the spectrogram for each detected vessel, both in the whole imaging plane. So the strategy we propose consists of using a clinical scanner to perform the two ultrafast sequence. Then, using the spectrogram, we can compute the resistivity index for each detected vessel by detecting the spectrogram envelope. Now, this spectrogram also provides information about the axial component of velocity, and with the flow direction, we can estimate the 2D flow velocity mapping from which we can compute the pressure gradient. Another approach to get information about pressure consists of estimating the vessel diameter variation from the ultrafast Doppler, and by knowing vessel thickness and stiffness provided by the elastography, we can obtain information about local pressure. Now, in the frame of this project, 3 series of acquisitions have been performed at the Necker Hospital, where a total of 12 patients have been recruited at the moment. An algorithm has been developed to obtain blood flow signals that adapts to patient renal acquisitions, this to get the power Doppler image. An additional filtering step was developed to further improve the power Doppler image, this in order to compensate for the TGC effects. And preliminary quantitative results of vascularization were obtained through resistivity and velocity mapping. Here you can see a typical result from the elastography where we can observe the stiffness mapping at a certain region of the kidney. And here superimposed with the conventional color Doppler. So at the moment, the elastography sequence provides spatially renal stiffness at a certain point of the cardiac cycle. Here you can see an example of the power Doppler image, here without and with TGC compensation, where we can see an improvement in vessels visualization. Here, 3 of the results that we can obtain from the ultrafast Doppler, the power Doppler image, the color Doppler, and the corresponding resistivity index map. These preliminary results were obtained, were evaluated by using clinical measurements. So here, it's a typical result from clinical, and here for our strategy. So in a qualitative way, we know that the new sequence improves visualization of small vessels, we can see here, and related to the resistivity index at certain points, as in clinical it's not possible to obtain a resistivity index map, we found relative error less than 10%. Here, result from the velocity, from which we can detect some challenges. One of them is the low resolution in the lateral component. At most of the time, the velocity only appears in the axial direction, and that the evaluation of the algorithm can hardly be done by patient acquisitions, as we don't know what is the true velocity in these cases. So at the moment, we can obtain a good power Doppler image for kidney and the corresponding resistivity index map. Right now, we are working on improving the method and strategy to get information about pressure. As the main conclusion of the work, an ultrasound elastography sequence was developed and applied on patients. An ultrafast Doppler sequence has been developed and further improved using patient acquisitions. Power Doppler results were improved by applying compensation for TGC effects, and preliminary results were obtained from patient vascularity and resistivity index, which were evaluated using clinical measurements. As a future works, we will find out if it's possible and how to improve and optimize the ultrafast Doppler sequence, for instance, by implementing vector Doppler approach and using a curve probe. We are also going to develop in vitro experiments in order to better evaluate the strategy we propose to get information about pressure. Thank you very much for your attention. I would also like to thank the École Polytechnique and Foundation Beta Kurzweller for their financial support, which makes the development of this project possible. Finally, it will now be a pleasure to hear your comments and questions.
Thank you very much, Lenin. So we are all expecting questions. So, yeah, I think, I know, okay. So congratulations, you received congratulations. It's really good.
Thank you.
So, yeah. So if there are no questions, we will move to the next speaker. So thank you again. Which is, who is Maëlle Wilbert, sorry, from the LOB at the École Polytechnique, and she's going to talk about in vivo assessment of corneal transparency via the analysis of images obtained by spectral domain optical coherence tomography in a clinical setting. So we can see.
Thank you. Thank you for the presentation. Can you see my screen?
Yeah, your screen and you also.
Okay, great. Thanks a lot for the invitation to present my work today. So I'm Maëlle Wilbert, currently second year PhD student at École Polytechnique in Laboratoire d'Optique et Biosciences. And my work deals with in vivo assessment of corneal transparency in a clinical setting. So first of all, why assessing corneal transparency? So as you can see here, cornea is the first lens of the eye, providing 2/3 of its refractive power. And the optical and biomechanical properties, including transparency, mainly depend on the thicker part of the cornea, which is called the stroma here. And nowadays, loss of corneal transparency as occurs with various pathologies, infection, immune reaction, trauma, aging, or surgery, are a major cause of visual handicap worldwide. And actually, the current means to assess this transparency are still subjective and qualitative. This is the case of the reference method in the hospital to assess corneal transparency, which is called slit lamp biomicroscopy. And authorities conduct a gross observation of opacities and give a grade to transparency on an arbitrary scale, which is hardly reproducible. So implications of objective corneal transparency assessment are diverse, such as early stage detection of pathologies, quantitative follow-up of patients during treatment or after grafting, and the optical quality control of eye-bank corneas. This project gathered researchers from very different fields as well as clinicians. I'm currently co-supervised by Garsten Plaman and Christina Hirsch. So this whole team proposed a novel method for corneal transparency measurement based on the physics of propagation of light in the media. It has been demonstrated ex vivo, and my current work is the transfer to clinical ophthalmology on in vivo images. So I'm now going to introduce how we get quantitative data from images acquired on a standard ophthalmic device. So as I said just before, we focus on the stroma, so the thicker part of the cornea, and it is composed of several types of structures at different scales that scatter light. And this scattering property makes us able to get depth-resolved images from the cornea, such as optical coherence tomography. You can see here typical images authorities have in the hospital here. And actually, when we looked at raw images, we identified two types of artifacts on the images you really had to remove in order to analyze only the signal that depend on tissue properties. So in a nutshell, just to give the solution for artifact removal, for this first artifact, column-like one that depends on a reflection on the top part of the cornea, I just, automatically detect it and crop it. And for the second artifact, which is a focusing artifact, there is a numerical step of flattening plus segmentation of the stroma. And then I designed a customized correction mask based on principal component analysis of in-depth OCT signal. This step is just a very little step in the preprocessing part of our analysis. And you really do not have to understand all of this. And my message here is that a few parameters derive from this full analysis, and those parameters are physically relevant since they characterize specific aspects of image or tissue, such as data quality, the homogeneity of the stroma, or the scattering of the cornea. So parameters may depend on each other. And the parameter for transparency here depend on the homogeneity of the stroma. And when stroma is homogeneous, that is to say bird ratio close to 1, well, we can define a parameter of coherent transmittance, which is the fraction of light, coherent light transmitted by the cornea. So now is the time for 2 applications in the clinical setting. First of all, we wanted to find the ranges of our objective parameters for normal corneas. So we got 85 normal corneas from 44 patients and had 2 images, OCT images per eye, acquired during clinical routine examination. And well, we checked our parameters for this range of people. Regarding data quality, we had good quality images all the time. Regarding stromal homogeneity, we expected to get homogeneous stroma since those cornea are supposed to be normal. And this was the case since this parameter was close in the order of magnitude of unity. And in the end, the parameter for coherent transmittance of the cornea was, in average, close to 51% for normal corneas. And while looking at correlation of parameters, we found no correlation between transparency and age or transparency and corneal thickness in this particular group, who is quite young, that may be said. And last application is the post-graft follow-up. This is work in progress, but I wanted to show you this histogram that shows the evolution of transparency with time after graft of people having had a Fuchs dystrophy of the cornea. And actually, when we look at literature, we learned that the maximal visual acuity is recovered 6 months after graft. And well, while looking at the data here about increase in transparency and stabilization, well, we assume that this method may unveil the contribution of corneal transparency stabilization to early stage recovery of visual acuity after a graft. So now it's time to conclude. So we transferred our method from the lab to the clinic. And regarding application, we defined the range of parameters for normal corneas, as well as applying this to quantitative post-graft follow-up. And regarding perspectives, where we have to quantify the robustness of the algorithm, find correlation with psychophysical measurement of vision, as well as extend the application. So for example, refractive surgery or optical quality control of grafts. Well, thank you for your attention. If you have any question, I will be happy to answer.
Okay, thank you very much, Maëlle, for this nice talk. So, are there any questions, please? No.
Okay.
Unfortunately, I'm not Abdul, able to ask always questions on any type of topics. But anyway, so thank you again. And now we are going to listen to the talk of Clotilde Raoult from the lab again on quantitative structural imaging of keratoconic human corneas using polarization resolved second harmonic generation microscopy. So we can see you and your presentation.
Thank you. Thank you for the presentation. I'm very pleased to present you today a part of my PhD work, which is about the structural imaging of keratoconic human corneas by multiphoton microscopy. This study was performed at the Laboratoire d'Optique et Biosciences located in École Polytechnique. So before to present you the purpose of the study, let's take a look at the structure of the cornea. The cornea is composed of 5 different layers distributed along the corneal thickness. And the biggest layer is the stroma. And this is this layer which confers to the cornea its mechanical properties and its transparency. The stroma is mainly composed of a superposition of collagen lamellae which run parallel to the surface of the cornea. And each lamellae is formed by thin collagen fibrils all aligned in the same direction. In some cases, the cornea is distorted and takes the shape of a cone due to a local thinning. This pathology is called keratoconus and provokes important visual impairment as irregular astigmatism and eye myopia. In the most severe cases, the patient needs penetrating keratoplasty, which consists on remove the cornea to replace it with one from a donor. So this pathology presents the shape modification, which is strongly linked to collagen reorganization. For instance, previous studies have shown that the lamellae in keratoconus present some ondulation and disorganization compared to healthy cornea, which have really straight lamellae. But these studies show some limitation. They are either qualitative or in a small field of view or on a very few number of samples. In this context, our works aim at quantifying these differences of organization with an automated and hand-based method. And of course, we want to push this study in all the corneal thickness and not only in the first anterior micrometer. So in order to access this information, we use nonlinear microscopy, which is also called multiphoton microscopy. This 3D imaging technique uses the interaction between several photons and the tissue to acquire images. It enables to access at higher depths in tissue and to perform multimodal imaging with a resolution similar to confocal microscopy. For instance, with the same excitation, it is possible to observe without any staining cells with two-photon excited fluorescence and collagen with second harmonic generation. The signal of second harmonic generation is specific to non-central symmetric structure, and fibrillar collagen exhibits strong signal because its peptide bonds, which act as nonlinear dipoles, are all aligned in the same direction inside the fibrils. So we use SHG to make polarization reserve measurement. By rotating the angle of the linear polarization of the excitation beam, we observe on each pixel a variation of intensity, which is linked to the orientation of the collagen fibril in the focal volume. So it enables to get information about the in-plane orientation of collagen in each pixel, and we can also calculate a parameter R², which filters the pixels that are not consistent with our theoretical model. It could be, for instance, pixels where there are some crossing fibers. With these three parameters, we can create an HSV image in which we can see the orientation as the color, the mean SHG intensity as the brightness, and the parameter R² as the saturation. In order to access to the quantification of the disorganization in keratoconus, we use 2 micrometer transverse section of 8 healthy corneas and 6 keratoconus, which are at the most severe stage of the pathology. The specificity of this transverse section are that they are embedded in HEPA, which is a hard material, and it helps to preserve the structure during the cutting. So to quantify the lamellae organization, we acquire different images through the corneal thickness, and we define 3 areas: the anterior, the middle, and the posterior part of the cornea. So just to show you the kind of image we obtain, here are the acquisition on the 8 control corneas and the 6 keratoconic ones. From this acquisition, we can qualitatively see that the control corneas show straight lamellae superimposed, whereas keratoconus seems more disorganized with different collagen orientation. This can also be seen by looking at the distribution of the orientation of the pixels. To quantify this distribution, we use the normalized entropy. This parameter will be equal to 0 for a distribution with only one direction, and it will increase with the disorder to reach 1 for an isotropic distribution. For instance, if we look at these 2 distributions, we can calculate the entropy and observe that the control cornea has a smaller entropy than the keratoconic one. When we look at the result from all the samples, we obtain significant differences in all the parts of the cornea. In every area, the entropy is higher in keratoconic corneas, which is consistent with the fact that the lamellae are more disorganized in this pathology. And this property is verified on all parts of the cornea, which is a new result. So in conclusion, this PSG technique gives us access to a direct and accurate measure of the collagen orientation on each pixel. Thus, there is no need of any image processing, which often require to look at several pixels and degrade the optical resolution of the image. This automatic method gives quantitative and best results with a large field of view. Concerning the biomedical result, this study have proved that the disorganization of the lamellae in keratoconus is observed in all the different thicknesses of the cornea. The quantification of this disorder in the middle and posterior part of the cornea is a new result that confirms that the disorder in keratoconus is all over the cornea, which is consistent with the mechanical behavior seen by ophthalmologists. So as a perspective, this method could be judiciously implemented in further studies to analyze other corneal structural pathology, as well as other collagen-rich organs that exhibit a highly organized structure at the micrometer scale. Before to finish, I would like to thank all the people who have contributed to this project. So my team in the Laboratoire d'Optique et Biosciences and our collaborator from Germany. Thank you for your attention, and I will be happy to answer to your question.
Okay, thank you very much, Clotilde, for this very good presentation. So are there questions, please? Yes.
So I have a question about the disorganization of the tissue of the lamellae. So is it involved only in the keratoconus situation or also in other pathologies?
There is some other pathologies, but so for instance, I think that foot dystrophy also exhibits some disorganization inside the lamellae structure. But I think that keratoconus is the most common pathology which exhibits this kind of difference.
Okay, thank you.
And me also, I have a question regarding the preparation of your sample. So how do you prepare your human cells to deposit it on a glass slide, or it is performed in liquid? It's not completely clear to visualize. How do you?
So we directly use human corneas which were taken from, so for control cornea is taken from dead people. And yes. For keratoconus cornea, it's when at the most severe cases, the cornea need to be removed. So we took this sample and they are fixed. And after they are embedded in this apron material, which is really strong. And it's cut in a really thin section and put in on a microscopic glass.
Okay. Okay, thank you for this precision. Okay, so thanks again. And now we will move to the next speaker, who is Matthew Gleason, again from the lab. And he's talking about multi-scale characterization of myelinated tissues using multiphoton microscopy.
So, hi, can you hear me?
Yes.
And can you see the screen?
Yes, exactly, also.
Great, perfect. So I'll crack into it. So, yeah. Hi, I'm Matthew Gleason, a postdoc at the I'm Matthew Gleason, a postdoc at the LOB, working on the brain harmonics project, or give it full title, multi-scale characterization of myelinated tissues using multiphoton microscopy, which is a bit of a mouthful, so I'll break it into 3 little sections, talking about myelin, multiple sclerosis, and where our project fits in, a little bit about laser microscopy and the contrast mechanism we're exploiting to evaluate the stage of myelination and demyelination in MS, and how we're using polarization and wavelength control to increase our imaging depths and specificity before some very short conclusions in future work. So myelin It's the name given to the lipidic sheet that surrounds axons in the brain. So it's essential for healthy conduction. It's about a micron thick. So in healthy people, you have a nice thick coating, but for multiple sclerosis, you actually have a degradation of this myelin sheet where it begins to disintegrate, where it gets to the stage, it eventually affects axon function, leads to neurodegeneration. Quite a widespread disease, but quite a widespread disease in Europe with a high cost per patient. So currently we use MRI technology to diagnose it, but it has low specificity. It's quite a different scale. So if this demyelination occurs over a micron and you're using technology limited to 50 micron cubes, not ideal. So we're evaluating the potential of nonlinear laser microscopy for multi-scale, high resolution, multifocal imaging. So I say multi-scale, I mean, we're trying to develop a microscope that can look at millimeter, hundreds of microns down to single myelinated tracks using nonlinear laser microscopy. So we have our long wavelength excitation, where we can combine several contrast modalities such as fluorescence to look at molecules, SHG to look at non-central symmetric structures, and then THG, which is what this project is centered on, which is ideal for looking at bases and lipids. So THG, nonlinear optical process, where you have a long wavelength excitation, 1500 nanometers, and the photons are upconverted into 500 nanometer wavelength. It's an interesting contrast mechanism, as if you have a homogeneous medium, you actually don't get any signal, which is strange, but there's a slight phase slip near the field of focus of the field, you get destructive interference. But for the case where you have an interface between two different medias, or geometry or susceptibility, the interference is partially constructive. So this is ideal contrast mechanism for looking at material change, such as white matter to gray matter, and highlighting interface, such as those found in myelination. So an example comparing THG to GFP in myelin in GFP tagged mouse brains, we can see here where we have THG and GFP over the first 5 layers of the neocortex. So looking at the overlap between the two, we know that THG is capable of imaging myelin without labeling and at a higher depth, and we can image both in-plane fibers, so say XY, and out-of-plane fibers, YZ. So the example of this is just shown here through the line scans, where we can see a higher contrast with better specificity for THG versus 2-photon fluorescence. So we can do this label-free and larger depths with better contrast for in-plane and out-of-plane. And seeing as we know THG can detect myelin, we might as well involve polarization into the mix, where we have an example of just probing a lipid onion here, where we can look at THG from the interface, THG from bulk, where this is an isotropic medium that's birefringent. You can do a Fourier analysis to determine the type of THG we're actually seeing before using a bit of MATLAB to extract lipid orientation. So we know we can look at myelin, we can resolve its properties using a polarization study. But if we want to talk a bit deeper, we can actually use polarization again to increase our maximum imaging depths. So an example of this is shown here in a myelin in a brain slice for a mouse brain, where we have cortex, corpus callosum in the interior of the brain. So if we align our polarization, our laser polarization perpendicular to the myelin, we can actually image a bit further down compared to having it parallel. So this is due to the increased amount of interfaces the polarization has to pass through, as I said previously, where you can actually use this polarization control to image a bit further down. So for an example here shown for 1150 nanometers excitation, we can actually increase our imaging depth by a couple of %. So we know how to get down deeper, but what we're currently limited at is the state of our laser system. So we'd like to do in vivo imaging eventually, so you can't really cut it in two. So an example of using longer wavelength excitation is shown here for 1.15, 1.3 micron, 1.7. So currently we can reach down to about 700 microns in depth before we lose our signal. Currently waiting on our new laser system, an OPA prototype collaboration with LCF, to see how much further down we can image. So I know this is very quick, but just as a summary, THG sensitive to sparse, in-plane and out-of-plane myelinated axons. We can use polarization and wavelength for deeper imaging. Currently I'm working on finishing the installation of some new equipment like an electro-optic modulator, spatial light modulator. So we can do fast polarization and wavefront shaping, and I'll apply polarization analysis, THG spectroscopy measurements. So it's short and sweet, but thanks for your attention. Thanks very much for the Foundation, Bet and Court Shooter, and the Polytechnique BME Seed Grant Program for funding this work. So, yeah.
Okay, thank you very much, Matthew, for your presentation. So we have time for questions. No questions? So apparently there are two, I saw two hands, but I don't see the name, Sylvia.
So it's me, so I have a curiosity. So if you can speculate, so which depth you think that you will be able to reach once you will have improved all the system?
So we're hoping to go sub 1 millimeter. So for mouse brains, which is the sample we actually test, cortex, so the yolk at the top, it's about 1 millimeter in depth. So we'd like to be able to probe fully down through this, but we're currently waiting on the delivery of a new OPA system that is slightly same wavelength, but a better pulse duration, so it's more optimized for THG. So hopefully in vivo we'll be able to go sub 1 millimeter. Together with the EOM, we should be able to go do that a bit further again. But you know, the joys of installing new equipment, it's always a slow process.
Yeah, it's a long process. Thank you.
Okay, do we have some others? Questions? No? Okay, so thank you again, Matthew. And now we come up to last speaker of this session, Hugo Blanc from the lab, and he's going to talk. Hello, so we can hear you. And so he's going to talk about large-scale volumetric multicolor microscopy of mouse brain tissues.
Okay, can you see my screen?
Yeah, we can see your presentation.
Okay, thank you. So hello everyone, and thank you for the opportunity, and thank you for the introduction. So yeah, I'm going to present you the project that I've been working on in the Laboratoire d'Optique et Biosciences, which is indeed about the development of large-scale multicolor multiphoton microscopy platform for brain imaging. So here we are developing microscopy techniques for neuroscience. And so understanding how the brain develops and how it is organized is a major goal for modern neuroscience. If we take the example of the mouse brain, which is already order of magnitude smaller than the human brain, it already contains hundreds of millions of neurons on top of being heterogeneous and scattering media. In the meantime, neurons usually present thin submicron-sized axons projecting over millimeter to centimeter long distances and across multiple brain regions. So brain-wide volumetric imaging at micron scale resolution is therefore required to capture neural cell networks. Finally, the brain is a very dense tissue, so contrast strategies are required to identify individual cells within their environment. For this reason, multicolor fluorescent microscopy can be very useful, as it is a key technology in biology, allowing to spectrally differentiate labeled cells and analyze their interactions. Among the available microscopy techniques, we are using multiphoton microscopy, which is one of the most popular, especially for tissue imaging. Since 2000, several technological developments have contributed to the maturation of the multiphoton microscopy technologies. So in linear one-photon fluorescent microscopy, the fluorophore molecule is excited upon absorption of a single photon, while in a nonlinear excitation process, multiple photons interact simultaneously with a single molecule. So for linear microscopy, fluorescence comes from the whole excitation cone, whereas in multiphoton microscopy, the excitation is confined within the focal volume, and this phenomenon is called optical sectioning. It generates significantly less out of focus, resulting in clearer image and less phototoxicity. As such, it is very well suited to image the brain, as it provides cellular to subcellular micron scale resolution upon hundreds of microns of depth within thick tissues, with the additional advantage of providing multiple contrast possibilities, as it was already presented in the previous presentation. With that being said, there are two main strategies to optically image the whole brain at the required resolution. With the first approach, the brain is imaged as a whole, and by chemically removing diffusive molecules like lipids and homogenizing the index of refraction of the structures, it is possible to image much deeper inside the brain. And this technique is called tissue clearing and index matching. The tissue clearing is usually pairs with the light sheet microscopy, which take full advantage of this approach. And with light sheet microscopy, the sample is illuminated with a thin sheet of light perpendicular to the detection objective. And the emitted fluorescent light is collected by the objective and projected to a fast capturing camera. And then by scanning the light sheet, you can image the entire volume. On the other hand, an approach requiring minimal tissue preparation is called serial section tomography. And it's based upon the methodology of alternating 3D imaging and physical sectioning in order to reconstruct large volume while keeping good resolution and contrast. It can be used with various imaging techniques, including, in our case, two-photon microscopy. It has been routinely used for 10 years in big neuroscience research centers like the famous Allen Institute. But while light sheet microscopy is much faster than point scanning based approach, thanks to the parallelized illumination and detection strategy, it is more sensitive to optical and chromatic aberration. Image quality and resolution is therefore higher for serial section two-photon microscopy, and it is easier to keep a good registration between color channels for multicolor imaging. On top of this, STP also requires only a minimal preparation, whereas the tissue clearing is a long and difficult process. At the Laboratoire of Optique et Biosciences, we recently developed a large-scale multicolor multiphoton microscopy platform. This approach is called CHROMS microscopy for chromatic multiphoton serial microscopy. And we use two-photon lasers with distinct and well-chosen wavelengths to realize two-photon microscopy. Then we precisely overlay the two laser pulse spatially and temporally, which allow them to interact with one another and generate a third excitation channel by two-photon excitation with wavelength mixing. The precise spatial registration of the three excitation channels is intrinsically guaranteed, which is one of the major advantages of this approach. Then we combine this multicolor imaging technique with serial section tomography presented earlier. And so each layer of the tissue will be successively imaged and sectioned. And finally, CHROMS platform is mainly dedicated to studying the mouse brain. So we worked in collaboration with Jean-Yves' team at Institut de la Vision, which designed transgenic mice expressing a multicolor labeling called Rainbow. For CHROMS acquisition, the brain is embedded inside the gel matrix and cross-linked with it to ensure good rigidity during sectioning. The acquisition of large volume is done by alternating acquisition, then sectioning of roughly 100 micron thick layer of tissue that we call sections. For every section, the transverse region of interest is covered by occurring successive image of the objective field of view in a snake-like pattern. And for each position, the objective field of view is scanned in order to create 2D image. And repeating this process at different depths allow to image small 3D volume composed of a stack of 2D images that we call a tile. With this approach, it is possible to tune the acquisition configuration from a histologic-like 2D acquisition with only one planned image for each section to a near isotropic volumetric acquisition. During the PhD of Lamia Abdeladim with the collaboration of the Jean-Yves team, CHROMS allowed to acquire several cubic millimeters of multicolor label mouse brain with submicron resolution and histologic-like whole brain acquisition composed of 2D planes separated by around 100 microns. However, this acquisition is very long, so it can take more than a week. So one of the goals of the project is to speed up the acquisition process. On top of the multicolor imaging, it is also possible to map large volumes of tissue using a THG contrast that was presented by Matthew, providing a wide range of useful biological information. And the CHROMS platform can also be used to image other organs. The main goal of my current work is to optimize the acquisition workflow to get a robust, reproducible, and automatic platform that can be used in semi-routine. And I am also improving the CHROMS platform to get closer to the maximal acquisition speed reachable with this approach by improving instrumental parts of the setup and the image reconstruction workflow as well. The main application goal is a volumetric multicolor dataset of all brain vision, especially the cortex and the hippocampus, with nearly isotropic and chromatic resolution. And other projects are ongoing as the THG imaging and imaging other biological tissues like the heart. So I would like to thank all the members of this great project at Institut de la Vision and Laboratoire d'Optique et Biosciences, and of course, IP Paris. Thank you for your attention, and I will be happy to answer questions.
Okay, thank you very much, Hugo, for this very nice talk. Once again, are there any questions? No, I don't see. There is no questions, unfortunately. So, okay, so this is the end of this session. And I think we can largely applaud our 10 speakers for the very high quality of their presentations. Really enjoy and hope that it was the same for you, the audience. So, so now we have the lunch break. And so we are on time. So probably, I suppose that we will start at 2 p.m. If Sylvia can confirm.
Yes, we will be back at 2.
At 2. Okay. So, and anyway, don't hesitate to use the Q&A option to ask questions to the speakers. Okay, so thank you very much and see you after the lunch. Bye.
Bonjour Clément.
Bonjour. Bonjour. Ah, je vais essayer de partager mes diapos alors.
J'arrête.
Et je vais fermer hop, tout ce qui traîne. Hop, et voilà. Alors, PowerPoint. Voici. Et là, je suis en mode présentation. Est-ce que vous les voyez?
Oui, c'est parfait.
Super. Alors, je vais juste essayer parce que j'ai 2 films. Je vais vérifier qu'ils marchent. Vous les voyez?
Oui.
Super. Eh bien, c'est parfait. Ok, eh ben, c'est tout bon pour moi alors. À tout de suite.
À tout.
À tout de suite.
Bonjour. Bonjour.
Salut Cédric.
Je peux éventuellement faire un test pour vérifier que ça marche aussi. Oui. Ouais,
Ouais, pour moi, ça marche.
ça marche. OK. Ah bon. Bon, bah, voilà. OK. Bon, bah, à tout à l'heure aussi.
C'est à l'heure.
on y va peut-être.
Oui, si tu veux, on peut commencer.
OK.
Je ne sais pas si Clément est... Ouais.
Je suis là. Je vais partager mes diapos. Hop.
Voilà, so je vais juste introduire. Okay, so welcome back to this BME Symposium. Thank you for all of you to be here. I am François, the head of the Laboratory for Optics and Biosciences. And it's my pleasure now to chair this session, which is about the laureates of the BME Seed Grants Program 2020. So just let me recall you that this program, which is funded by the Fondation Bettencourt-Schwiller, aims to help a duo made of a scientific researcher and a medical doctor to launch an innovative research project with potential medical applications. And this year there were 18 proposals, and 3 of them have been selected by the international committee that was presented by Abdul this morning. And now we will listen to these 3 projects. And so the first laureates are Clément Campillo and Elsa Labrune. So Clément Campillo is associate professor in Université d'Evry and is a specialist of the mechanics of the cell cortex that he studies on living cells and reconstituted actin membrane systems. And Elsa Labrune is a medical doctor specialized in the reproductive medicine and biology, and she works at the Lyon Hospital in the reproductive medicine department. So the project is about, as you can read, microfluidic selection of oocytes for reproductive technologies, and it's Clément Campillo who represents the work. So Clément, you can for 15 minutes.
Thank you very much for the introduction. So I warmly thank the organizer of the program and its funders to have selected our project. We are very happy to have the opportunity to start this project. So we are actually 3 parts, myself as a biophysicist, Elsa, who is a medical doctor, as you just say, and also we work with Marie-Émilie Terret, who is in Collège de France, and who is a specialist of the cell biology of oocyte division. So it's really an interdisciplinary project. Rose Bulto is an engineer that has been hired thanks to the BME grant, and Lucie Barbier is an engineer in CIRB, and both of them have done the experiment that I'm going to present you today. So our question. So we study female meiosis, so the formation of sexual cells in females. And this is an error-prone process. So that means there are errors in the formation of this cell, in the segregation of their chromosome, and this phenomena increases with age, as you can see in this plot for trisomies. So this is a major public health problem. In France, you can see that 10% of couples are infertile. 3% of babies are born using RRT, so assisted reproductive technology, but the success rate of RRT is only 17%. And so that means it costs 300 million euros to the social security each year. And it's getting worse and worse because today 25% of women are over 35 when they gave birth. So they are in this part of the curve where the risk of aneuploidies is very high. So the question we want to tackle is how can we access non-invasively the quality of oocytes for in vitro fertilization and more generally RRTs. So we started this project by working since 10 years now with Marie-Émilie Terret on the link between meiosis and cell mechanics. So the female meiosis relies on 2 asymmetric divisions that form 2 degenerating polar bodies. You can see one here and one big haploid cell that you can see here that will be fertilized. The asymmetry of this division relies on the off-center positioning of the chromosomes inside the cell that you can see here. And at this step, this movement of the chromosome is actomyosin dependent. Actin is one major component of the cell cytoskeleton. Okay, and this is in mouse. Okay, we are working in mouse with Emily. So what Emily shown is that at the moment where the chromosomes are moving towards the periphery of the cell, there is a thickening of the actin cortex that you can see here. You have a very thick actin cortex, and we started working together by trying to understand what is the role between the mechanics of this cortex and the asymmetry of the division. So what we have done is to treat these ovocytes as liquid droplets. And liquid droplets in particular have a surface tension, and we measure the cell tension, the tension of the periphery of these cells. And to do this, we aspire the cell inside glass micropipettes, as you can see here. What we have shown is that this thick cortex was excluding myosin motors from the actin cortex, leading to a drop in the tension of the cell. Then we have shown that manipulating this tension, we were highlighting a mechanical checkpoint. This means that the tension of the ovocyte has to be between well-defined values of tension. If the egg is too stiff or too soft, the asymmetric division won't happen properly. Finally, we have shown that after fertilization, the tension re-increases to go back to symmetric divisions for the first divisions of the embryo, and also that aberrant tensions were leading to aneuploidies, defect in the segregation of chromosomes. So very importantly, that shows that there is a link between cell mechanics and cell division. So this is where we started working with Elsa to see if ovocyte mechanics could be a biomarker for the selection of human oocyte in the context, in a therapeutic context linked to IRTs. So what we have been doing since the beginning of this collaboration is first use atomic force microscopy. An atomic force microscope exerts a known force here on a tip that you can see and indents inside a living cell, here an ovocyte, and you can measure this indentation. What is interesting for us is that AFM has a higher throughput than micropipette aspiration. It's less invasive for the cell, and you have the possibility to retrieve the oocyte to follow its development after the experiment. So Rose Bulto has shown that you can record force versus indentation curves on the ovocyte at different stages of the meiotic development. These ones are the early, even immature. This is the beginning of the meiosis, and this is the end. And what you can see is that the cells are very stiff at the beginning, and they get softer and softer through the course of the meiosis. By analyzing this force curve, we can measure the tension of the cells at each stage, as you can see here. And very interestingly, we can see that tension follows the same trend than the value that we have measured years ago with micropipette. So we have a new tool that we will use to screen different mutant oocytes, in particular extra soft and extra stiff. And this will be very helpful to calibrate our microfluidic experiments. And now that these experiments work, we are going to try them on human oocytes in the lab of Elsa Labrune. And now the core of our project is the development of a microfluidic screening of oocyte mechanics. So how do we do that? First, we have designed a chip, a PDMS chip, a standard microfluidic chip that you can see here. The liquid is in blue. And on this chip, you have an inlet where you can put an oocyte. You have another inlet in which you can push the oocyte by a fluid flow, a controlled fluid flow, and then the oocyte pass through a constriction that you can see here. And finally, there is an outlet where you can retrieve the oocyte and take it, hopefully, to be fertilized. So I will show you our preliminary results with this microfluidic chip. And as you can see here, these are results that we have on a mouse oocyte. In this example, you have a prophase oocyte, so that means very early in the meiosis. And as you can see here, we are able to put them in our microfluidic to bring it to the constriction here. And as you see here, we are increasing the pressure with which we are pushing the cell to go through the constriction. First, it is stuck. We increase the pressure, and soon you will see that we reach a pressure that will be enough to push the ovocyte to the constriction just right now, okay? And we can measure the threshold pressure at which it enters. On the other hand, if we take an ovocyte that is in late meiosis, so much later in the meiosis, we know that these ovocytes are much softer than the prophase one. You can see here that the pressure that you need to drive the ovocyte to the constriction is much lower, 2 millibars, and then it's gonna enter the constriction. And that is quantified here. You can see that this threshold pressure is different at different stages of oocyte development. So we can discriminate the mechanics of these oocytes using this type of microfluidic constriction. This is very useful because that's a technology that you could imagine bringing to a therapeutic context, and that could be used in any clinics doing IRT. And very importantly, we have checked that the oocyte, after this microfluidic experiment, will still develop properly and could be used for in vitro fertilization, for instance. Okay, so to wrap it up, we have developed a technique based on AFM that provides us mechanical characterization of murine oocytes so far. And of course, we want to transfer it to humans. In the next months, we will try to do AFM experiment on human cells in the lab of Elsa Labrune. And we have developed a chip, a microfluidic chip that is adapted to mouse oocytes, as you have seen just before. And now what we have to determine is what are the most accurate predictors of oocyte quality. Is it the entrance time in the constriction, the threshold pressure, or any other type of parameters that we will investigate? Then we will, when this will be a bit clearer, we will adapt our setup to human oocyte and bring it to the lab of Elsa. And we will have many problems to face, like sterility, oocyte retrieving, because we want to keep these oocytes. They are very precious. They come from patients, and they are meant to be used for in vitro fertilization. This means also that we have ethics concerns. We have to be sure of the consent of the patient and this type of aspects. And finally, we will correlate the mechanics of the oocytes and their quality. That means, will they develop properly, and will we have a pregnancy at the end of the process? We will correlate this information with many clinical data that Elsa studies, like age of the mother, tobacco, other diseases, and also do a single-cell RNA-seq to study which genes are activated and how they correlate with the mechanical properties of the oocyte. And I think I'm going to stop here, and I'd be very happy to answer your question, and thank again to the BME program.
Okay, thank you very much for this nice talk. So for the question, I propose that you raise your hand, but right now there is a question by Cécile Sykes. Maybe Cécile, you can use also the microphone. Sylvia, tu peux donner la parole à Cécile. Okay, Cécile, I think you can just switch on your microphone and put your question.
Okay, can you hear me?
Yes.
Yeah, so it's a question I've asked many times in these sorts of experiments. So I have some confusion between tension, stiffness, and AFM force. So how do you get one and the other? Can you clarify so that I never ask this question again?
No, that's a complicated issue. The good thing is that in AFM, a stiff material will give you a signature that is different from a tensed material. And to say it very clearly, the force indentation curve are parabols for an elastic material, and they are linear for a tensed material. So in our curve, we can see the two contributions. Practically, we fit our curves with x square and x, and we see the two contributions, one from stiffness and one from tension. So we can really separate these two contributions.
Okay.
Okay, other questions? In fact, I have one. You said that so the oocytes must pass through very narrow microfluidic channel. You said it was not a problem, but are you sure it's not a long-term problem for the oocytes which will develop normally afterwards?
Yeah, so as you see, we use constriction that are less than 50% of deformation, 50% squeezing. And because we can retrieve the oocyte, we can then take them back to the culture room and just follow hours after hours. And we have checked that they really develop as control oocytes. So
okay,
this is a strong point for this approach because the oocytes are very fine.
Yes, very, very nice. Okay, are there other questions? Apparently no, so thank you again.
Thank you very much.
Just a question by Roth Essier. I will put the question myself. How much of the variation is due to intrinsic stiffness and how much due to cell size?
Oh, the size is really conserved at like less than %. So the size is always the same in oocytes. They are super controlled. So the difference will be only due to the stiffness. But the size is not the same between mouse and humans. This is something that we will have to treat.
Okay, so I think it's okay for Ross. Other questions? No, I don't see. Okay, so let's switch to the second project, the second laureate. So there are Cédric Bouzig and Bruno Lina. So Cédric Bouzig is assistant professor in biology at the École Polytechnique and laureate of the INSERM Excellence Chair, and is head of the Nanoimaging and Quantitative Biology Group in the Laboratory of Optics and Biosciences. Professor Bruno Lina is head of the virology department at the Croix-Rousse Hospital in Lyon, and he carries out research at the International Institute of Infectious Agents, and is also a member of the French Conseil Scientifique. So together they want to develop a new nanoparticle-based ultra-sensitive fast COVID detection assay. It's a bit close from what we heard this morning. And it's Cédric Bouzig who will present the talk. Cédric, you can go.
Okay, thank you, Francois. I hope you can hear my voice and see my slides.
Everything's okay, yeah.
Everything is okay. So thank you, Francois, and thank you, everyone. So I'm really happy to present you the NASCAP project. So first, because obviously it's a project that is nicely funded by the Data for Cell Foundation. And secondly, and maybe most importantly, because it's very nice to present the project in front while such a fine assembly. Okay, so the NASCAP project stands for Nanoparticle-Based Fast COVID Detection Assay. So it's a partnership between the LB, myself, and Thierry Gacquin at the PMC at the Technique, and Bruno Lina at the University of So, well, the acronym is like it's now self-explaining. So basically, I'm going to be very quick to say what we like to want, and then what to do, and in fact, we want to develop efficient assays to detect COVID, and more generally to detect viruses based on nanoparticles. Okay, so the question is, how is it possible to diagnose infectious diseases, and notably viral infectious diseases? So as most of you must know, well, basically you can either detect protein, specific viral protein, or detect nucleic acids, for example, RNA in the case of SARS-CoV-2 detection. So, well,
Cédric, on t'entend pas très bien. Est-ce que tu peux peut-être rapprocher ton micro?
Yes, is it better right now?
Ouais, ça, je crois que c'est mieux.
Okay. So I hope it's going to be fine until the end. So, well, basically, how is it possible first to detect proteins? So, well, mostly you have two different methods, either ELISA method, which are in fact the gold standard for mass detection, which is, for example, massively used for detect HIV. And these techniques are sensitive, but well, they have major drawbacks that slow. It needs a couple of hours to be efficient, and they're usually performed on automat, on large platforms, and can provide results typically in a couple of days or one day. On the other end, well, most of you are, I guess, are familiar with them now. Lateral flow assays, presented by Fanny Musso this morning, are widely used for SARS-CoV-2 detection. Top of there, small foods that you can find in the streets now. But the other major advantage of this test are, in fact, really fast. You can have results in a couple of minutes. But there are major drawbacks. It's that they're net sensitive, meaning that typically, in the case of SARS-CoV-2, 50% of infection are in fact missed by antigenic tests. So on the other hand, RNA detection are being widely used either in labs or for diagnosis purposes to detect, in fact, viral material. You have, I would say, two different kinds of approaches, either purely hybridization methods that just rely on hybridization of a specific probe to a target, which has been the basis for now old methods such as Northern blots and fish. These techniques, in fact, have usually a low sensitivity. They can have very high backgrounds, and in the case of fish, so fish is a fluorescence hybridization in situ. They relies on the use of microscopes. So in fact, they're research techniques. They cannot be usually used for, in fact, diagnostic purposes. On the other hand, amplification-based methods such as PCR or isothermal amplification on the gland, are the most sensitive techniques that can be used for diagnosis, but they have major drawbacks. So that in fact they can be slow, and they're still lab techniques, meaning you cannot do that on the street, and you cannot do that on a simple bench. And another drawback is that they're all difficult to multiply, which is in fact a problem, for example, in the case of SARS-CoV-2, the detection of variants, meaning that it's highly difficult to detect multiple features of multiple variants only by PCR, and very often sequencing is mandatory to perform duty, but sequencing is very low technique, so it's not suitable for diagnosis. So this brings to our question is in fact how to really test a group and obtain results in situ within 30 minutes. By a group, I mean, like our current assembly, we could have said, we're not going to do our meeting by Zoom. We're going to have, in fact, a real physical meeting, but we like to be sure that everybody is negative before entering the room. So what we like to have a reliable test, meaning that its sensitivity should be comparable to PCR. We should be able to test rather quickly a typical group of 100, between 10 and 100 persons, at a classroom or while plane or train or wagon train. And we like to have results that may be quantitative and they can account for potentially different pathologies. We can project beyond the other case of SARS-CoV-2 or variants. And we like to do that in situ, meaning, it's not possible to, in fact, take samples on everyone and send this to a lab, even if you are in a place where we have labs. So we like to have a portable device, meaning that, in fact, this doesn't exist so far because, PCR is not a portable device and antigen tests are difficult to consider as very reliable tests. So, in fact, our objective is to propose an amplification-free, multiplexable RNA detection for in situ tests. So we have, in fact, two leads in this objective. In fact, can we transpose some of the techniques that require a microscope into a portable device? One question would be, that major problems of limits sensitivity of techniques, the fact that labeling agents are often not efficient enough for having, in fact, a good sensitivity. So this is the reason why we have started to use luminescent nanoparticles that have been synthesized by the team of Chirigato and Ecolpolitic. So this particle, as example, we have there, in fact, are really lanthanide-based nanoparticles that are doped by an ion which can be luminescent. Cases like presented here, you have luminescent uranium that can be excited blue in UV, and the banana image, as you can see on the spectrum in red. Okay, so what are the major advantages of this kind of probe? Well, first one, they are limited. Okay, but one major advantage is that they're photostable, meaning that they can be excited and can luminance, in fact, for arbitrary period of time. Meaning that there is no such process as photo bleaching for this particle, meaning that you can excite strong as you want this particle without causing photo damage on losing the fluorescence. Well, in fact, they're quite easy to image because of a large state shift. One striking pattern is that the fact they're a very long lifetime. The excited state has a very long lifetime. I will come back to this one, which is typically a couple of hundreds of microseconds, which is significantly longer than any other fluorescence. Another advantage of these particles is, in fact, with the same kind of matrix, in this case, validated matrix, it's possible to use different doping ions, and different doping ions means different colors. So, meaning that, it's possible to have a system where, we'll come back to that, you can excite simultaneously different nanoparticles, right, for targeting different fragments and having different colors, and so that it is possible to make them. Okay, so when we started the project, we started the project, in fact, on the left, where saying, these particles are efficient optical probes for biological application, and we have been able to show there the example that you have in the image, which is on the left. It's possible to detect single nanoparticles on regular microscope with a sensitive camera. But obviously, this is not, in fact, even if you can detect single molecules, it's costly because they are quite efficient microscope, and it's not a diagnosis method. I'm not going to propose you to take a sample, and install my microscope in front of a classroom. Okay, so the question we have is how is it possible, in fact, to maintain some kind of high performance, but in a system which is usable for bioanalysis and possibly in possible format. So, the idea that we have is it's possible to detect nanoparticles that are tethered to a surface, in fact, by a specific way. I'll come back to this point. So the idea is quite naive, is to say, we can excite these particles, and in fact, we're not limited by the amount of power that we can use. So we propose some kind of system which is called a dirty tube. So the dirty tube is, in fact, an event, not an event, but a system in which, in fact, the most part of the sample that has been excited is close to the surface. So it's possible to, in fact, high signal to detection. And we can use high illumination intensity because there is no fluctuation. It's possible to improve the system by, in fact, switching to real curve, so that it's possible to, in fact, to think about the system that does not require any rinsing steps. That could be, in fact, in terms of manipulation, very good. Okay. So, in fact, we converted these ideas in a system sense to major helps of the technical team, Jean-Marc Santes and Jefferson Salinas. So you have, in fact, our first prototype there on the left, where you can see a very simple system running on, in fact, fiber laser, in fact, photon layer that detects the light. Okay, so we can have illumination power, so we can detect a low number of particles, which is the objective of this system. So the question that we have there is, does it mean that necessarily that if we can detect a low number of nanoparticles, that we have time that it's very sensitive? So not necessarily true. Why is that so? It's first because, we can have, in fact, parasite symbols, meaning that if you are using high illumination power, you can have strong background or autofluorescence of any part of the system, solid substrate or liquid media, and whatever. It's also possible to have non-specific band. So my preference would be to show you how we can overcome the first limitation, in fact, by eliminating, in fact, any background or autofluorescence by using time representation. So, in fact, we've tested this aspect on a model system, which is, in fact, an immunoassay on insulin detection. So we use the concept of sandwich allograft for insulin detection. We use our system at high illumination power and dotted configuration, and we use modulated illumination, draw a shoulder. So what happens? Okay, if we modulate illumination, we have obviously modulated illumination. But what is interesting is the fact that due to the very long lifetime of excited state nanoparticles, in fact, we can separate. We can separate between, in fact, fast emission of other fluorophores. We can, in fact, eliminate the potential direct observation of the laser and just detect the slow component, which is due to the emission of nanoparticles. So it's possible to specifically extract whatever the illumination power is in fact, the specific nanoparticle signal which is due to nanoparticle. Okay, so what can we do with this? Well, we can detect insulin, which is not that interesting, but it's a model system. And we have seen that either in protein simulation or just by removing background or time illumination, it's possible to enhance detection limit that is usually achieved by ELISA with the various system by a factor of 1,000, which in fact corresponds to density on the surface of nanoparticle of typically 1,000 per millimeter square. So what does it mean? It means that in fact we can detect what we call macroscopic signal. What are macroscopic signals? Macroscopic signals are signals that usually require a microscope to use. Okay, so where are we now? So the idea is to, in fact, to translate this approach to the detection of RNA according to different possible approach. One on the top, which is in fact directly derived from the northern blot approach. The idea is to tether a capsule oligo on the surface to then to have your sample enter. In fact, you're going to hybridize with your capsule oligo. And then you have nanoparticles that have been coupled to a different oligo, which is also complementary of the target sequence you would like to capture. So at the very end, you have nanoparticles attached to the surface only in the case where your target RNA is present. Another approach is called fish-like approach, is to say, well, you just make a deposit of biological sample of cells, for example. You can imagine from an oral strapping, and then you just sterilize the cell. And then you have the nanoparticles that are complementary with another, which is complementary of target sequence of, in fact, the viral RNA that is in the cell. And once you have this construct, well, we can perform our ultrasensitive synthesization through our portable device. Okay, so while this is somehow the plan where we would like to go, in fact, I'm gonna be very detailed on each square of it. It's gonna take only 10 minutes for each square on this slide. No. So, no, well, in fact, no, I'm not going to detail, but just to explain the principle that we have. In fact, we are there, point where we are, in fact, we're looking at this model DNA system, meaning that we only detect, in fact, model sequence, either in the northern approach or by the fish approach, and then we're going to increase, in fact, the complexity to, in fact, at the end of the project, be able to make detection first on spiked samples, meaning in samples in which, in fact, a real RNA has been added, and at the very end, detection on real samples. And the idea would be that we make a real benchmark between PCR and antigenic test on our test. In fact, to identify what is the actual performance that we can achieve in a given amount of time with our test. Okay, so what we'd like to have the device. So we'd like to enhance maximum, in fact, sensitivity of it. So we plan to increase, in fact, the excitation by using a UV excitation to have the maximum button and have better sensitivity. So we plan to develop multiplexing. So the first idea is to perform in fact, spatial multiplexing, working well on a chip or for example, on well, on typical 96 well thing. So by optimizing nanoparticles, first the question will be to enhance again the stability, to optimize the brightness, to enhance sensitivity, and to use a multicolor in fact to have a possible spectral multiplex, either to detect multiple samples or, for example, in the case of SARS-CoV-2 mutated virus. Okay, so what I'd like to have at the very end would be a device which is portable and it's suitable for multiplex detection on a chip or in a stomach plate. Okay, so what was the consequence of this? If we're able to do that, we'd like to make the detection in less than 30 minutes at high sensitivity, when sensitivity which should be comparable to PCR, or at least better than regular antigenic tests. Or to possibly identify different variants in the case of SARS-CoV-2, which is the real problem now. But we like to make we may need to make a trade-off between the ultimate sensitivity and the speed at which we would like to have the testing. On the other hand, well, we plan to run industrialization if possible of this system so that we can distribute it for real tests in real life. And this could have major, well, say, biomedical application because of the general framework to detect any RNA virus that could be, well, sensitive and portable, meaning, well, you could make tests at a given group and have available answer before all these people are going to meet. So I'm going to end there. Time. Just mentioning the people that are involved in the project, either at ADB and at the PLC, the team at the and at the VFAT lab, you know, lab is a reference, in fact, in terms of detection of respiratory disease, and we are very involved in benchmarking different tests that we may be able to develop. I thank you for your attention. I will be happy to answer any questions.
Thank you very much, Cédric. I already see a question by Emmanuel Brouter.
Yeah, can you hear me?
Yeah.
Yes.
Yes. So hi, Cédric. Nice talk. I have two well, very technical questions. First, what is the size of the portable prototype that you show in your slides? And the second question is, what is the laser source inside that?
Okay, so the first portable device that I presented is not that portable because, well, it can still, well, a typical dimension is, let's say, 50 by 50 centimeters.
Okay.
So it's possible, well, but it can be miniaturized. But, well, the idea is not to have, in fact, an individual device, but to have, in fact, a transportable device that, for example, that could be like put on a bench or put on a table. That you can imagine. So it's not an individual system. Just the second question. Well, so far we have used, in fact, a diode-based laser, 466 nanometer 1 watt laser.
Mm-hmm.
But, well, as I briefly mentioned, so, well, the idea is to switch, in fact, to a regular illumination system for having UVC illumination. Because, well, absorption of vanadium particles in UV is more significantly higher than visible. By significantly, I mean it's 10,000 times higher. So
okay.
you can excite very strongly. So, but, well, it's I mean, so, but, well, the background will be considerably higher by using UV sources. So that's why what time resolution will be limited.
Okay, thanks.
Okay, are there other questions? Cédric, can you comment a little bit? How would you do with your device for detecting the variants of COVID, for example?
Okay, so the idea is to, I would say the easiest way is to use, in fact, spectral multiplexing, because, well, in fact, since you can use different type of nanoparticles with different doping ions, you can have, in fact, a couple of colors. I presented only two, Symposium and European, but it's possible to have. We know so far that identified with Thierry's team that we can have like 5 different colors that can be inserted into the same matrix. And this is one advantage of, in fact, of UV illumination, because UV illumination, in fact, is going to excite only vanadate, and you're going to have an energy transfer to the doping ions. So you have the same excitation, but you can excite all the different colors. So our system would be a mix. But in fact, the standard approach that has been used for, in fact, for fish, when you have a mix of different oligos, you have 5 different colors, meaning that, in fact, the resulting color is the signature of the configuration of your sample in terms of variance. Which 5 is not, doesn't seem a lot, but it's twice, more than twice that regular PCR chemistry. Because this is something which is widely used in France, because You may know if you are COVID positive, you are performing what is called a screening PCR, the screening quantitative PCR, in which you can discriminate between two different variants, typically beta and gamma, so alpha and beta variants. And if you want to know more, you have to sequence, and sequence is very good for population in vitro, but it's not suitable for diagnosis because you're going to have your sequence. If you want to sequence, you're going to have your answer like 10 days later, which is not good if you want to have a COVID diagnosis.
Okay, thank you. Other questions? No. So thank you again, Cedric. And we will switch to the third laureates, who are Abdul Barakat and Catherine Poirot. So I think I don't need to introduce Abdul. Everybody here, of course, knows Abdul very well. I know he's no longer there, but I wanted to take this opportunity to thank him for putting together this very exciting biomedical engineering program, because it is really at the heart of all this program. So you know, Abdul is a biomechanics professor at École Polytechnique and researcher at LADIX. And he works on fluid mechanics for arterial and mass transport, vascular cell engineering, and so on. Catherine Poirot is a medical doctor and an INSERM researcher director working in the Institut de Myologie in the hospital Pitié-Salpêtrière in Paris. And she's a specialist of the effect of mechanical stress on the skeletal muscles. And together they present a project about nuclear cadging on microgroove substrate, but this project will be presented by Claire Leclèche, who has been a postdoc in Abdul's group and who will benefit from the seed grants. So Claire, it's yours.
Okay, can you hear me fine and see the slides?
Yeah, here's perfect.
Okay, let's go then. So yeah, I will present you this project of nuclear caging on microgroup substrates, for which we were awarded the BME seed grant. And so, as you mentioned, so I'm developing this project in the team of Abdul Barakat here in Paris and in collaboration with Catherine Poirot from the Institut de Myologie. And so I will start by explaining a little bit how this project was born in the lab, because it's a bit different from what we usually do. And so in the lab, we're interested in the vascular endothelium, which is the cellular monolayer lining our blood vessels, which is composed of vascular endothelial cells. And we are interested especially in the shape of these cells. They can be different in vivo. You have regions where the cells are aligned and elongated, and regions where the cells are more round. And one of the reasons we're interested in this shape is that it has been associated with different sensibilities to the development of cardiovascular diseases. So there is a strong relationship between the shape and function of these cells. And so different factors can influence vascular endothelial cell shape. So you can see here a cross-section of the vessel wall with the vascular endothelial cell, which are subjected to the blood flow on their apical surface. And it has been shown in vitro that a laminar flow is able to elongate and align these cells. But more recently, the influence of the topography of the substrates on which they are anchored was also investigated. So as you can see here, they reside on surface called the basement membrane, which presents a certain structure, topography, at the micrometric and nanometric scale. And this is the kind of cue that we are interested in. To study this aspect, we are working in vitro with macrofabricated substrates composed of parallel arrays of micrometric grooves made in PDMS, the surface of which we coat with adhesive protein, and then we are able to culture human vascular endothelial cell at the surface of these microgroove substrates. Here you can see how a typical experiment looks like. On the first line, you see a controlled flat surface without any structure. And you can see that here the cells are color-coded with their orientation. And after the initial cell seeding, the cell proliferates and form a monolayer. And on the groove substrate here on the bottom, you can see the same dynamic of formation of the monolayer, but this time the cells align along the groove direction. And so using this substrate, we've been able to show that actually the dimension of the groove can modulate this alignment and elongation of the cells along the groove axis. And in particular, we've been able to show that increasing the groove depth from 1 to 6 micron significantly enhanced the alignment and elongation of the cells. And so the cell shape was quantified using this type of immunofluorescence images, where you can see in red the cell outline and in blue the nucleus. And so while doing this experiment and specifically looking at the cell nucleus, we realized that on the deeper groove, we could see the appearance of like very elongated nuclei, which kind of caught our eye. And so to see better what was happening in this configuration, we did some 3D reconstruction, which allowed us to realize that these very elongated nuclei I was mentioning were actually like completely confined and trapped into the groove. And so we call now this configuration the cage nuclei. And so we decided to study this nuclear deformation further. And so I'll make a little break here to explain you why we are interested in nuclear deformation in general. So the cells in our body are usually navigated in a crowded environment. So they experience confined migration. And it's, for example, especially true for the immune cells, which are patrolling in our tissue and need to like deform a lot, for example, when they are passing through the vascular endothelium or through inside the lymphatic vessels. And it's also true for cancer cells. And the nucleus being the biggest and stiffest organelle of the cell, it's like the limiting element in this confined migration. And as such, it has been the subject of active research. So there are different systems that have been used in vitro to study nuclear deformation, including, for instance, micropipette aspiration, or microfluidic systems with constriction that is deforming the cell and the nucleus, a little bit as the one that was presented before. And so the problem with those systems is that they still include some experimental complexity and they are quite low throughputs. And so in this context, we believe that our microgroove substrate has really a high potential to become a simple and high throughput system to study cell and nuclear deformation. Okay, so now I will present you some of the results that we already have on this project. And the first interesting observation that we made was actually by looking at the cell and the nucleus live. So here you can see a movie where the nucleus is stained in blue. And you can see that actually this caging I was mentioning is not permanent, but rather the cell and nucleus is able to go in and out of the groove. So part of a dynamic cycle of nuclear deformation. And in relation with this idea of like dynamic cycle of deformation, we were actually able to describe many different intermediate nuclear configuration in between the fully caged nuclei and the fully free nuclei. Okay, so then we try to quantify a little bit more the nuclear deformation induced by the caging. And here you can see on this immunostaining images with the staining for the nuclear envelope that the caging is actually subjecting the cell to like a lot of changes in the three-dimensional cell shape. And particularly, we've been able to measure that on average, the cage nuclei have a 30% decrease in their area and a 20% decrease in their volume, which is a quite significant deformation. The next thing that we addressed was the mechanism leading to this caging and nuclear deformation. And the first kind of most intuitive thing we've been looking at is the cell membrane deformation. And here you can see on this electron microscopy image that the cell membrane is really able to deform and adapt to the groove contour and to reach and adhere to the bottom of the groove. And to test if this was necessary for the caging to occur, we perform an experiment where now we put the adhesive protein only on the top of the ridges and not on the edge and bottom of the groove. And in this condition, we completely lost the caging and the nucleus are all like suspended now, which shows that the membrane deformation and adhesion at the bottom of the groove is necessary for the caging to occur. Then we investigated the cytoskeleton organization around the cage nuclei. And so here you can see the localization of the intermediate filament, the vimentin, in red, and the microtubule in green. And we've been able to show that these two cytoskeletal networks predominantly localize above the nucleus. In contrast to the actin cytoskeleton that was like strongly associated with the cage nuclei as stress fibers, but predominantly localized above and on the side of the nucleus. And preliminary experiments using actin depolymerizing drugs show in this case a decrease in the percentage of cage nuclei, which indicate that the actin cytoskeleton is a major player in the caging process. And so here is today our current working model of the caging mechanism. So the idea is that when the cell is placed on the groove, the cell membrane will spread and explore. It will deform and reach the bottom of the groove on which it can establish adhesion with the substrate. These adhesions are connected to the actin fibers, which are themselves connected to the nucleus. And this can pull the nucleus inside the groove. And we are currently investigating the role of the other cytoskeletal elements in this process. So now what is next with this project? So the next big step, and for us maybe the most interesting that we want to address, is to understand the link in between this nuclear deformation and the mechanical properties of the cell and the nucleus. And so to do that, we are performing AFM, atomic force microscopy measurements on cage and non-cage nuclei to understand if the process of caging is associated with changes in the mechanical properties of the nucleus. And we can also, and we will also modulate the mechanical properties of the nucleus using drugs targeting the chromatin compaction. And so finally, why are we so interested into this, the mechanics part of this project? It's because it's now more and more clear that the nuclear mechanics is involved in many diseases. One of them are laminopathies. So laminopathies are rare genetic disorders, which actually have a lot of different clinical manifestations. So such as cardiomyopathies, muscular dystrophies, neuropathies, etc. But they all involve a mutation in the lamin gene, which is the main component of the nuclear envelope. And so you can see here on these images, nuclei with the mutation in the lamin that present an abnormal structure, and they also show abnormal mechanical properties. And we can also mention that the mechanical properties of the cell and nucleus is involved also in cancer. So the nucleus in cancer presents different mechanical properties that can participate to their invasive potential. And so the idea for the future of this project is to test the behavior of this pathological cell in our microgrooved system. And so in particular for the laminopathies, we are collaborating with Catherine Poirot, which is a specialist of the diseases at the Institut de Myologie de Paris. And so she'll be able to provide us with cells from patients that we will test in our microgrooved system. And so in conclusion, we really believe that this new microgroove system will really help us to understand further the process of cell and nuclear deformation and mechanics, both in physiology and pathology. And I will end by acknowledging all of the people involved in this work. And of course, a particular thanks for the Fondation Bettencourt for founding this project. And thank you for your attention.
Thank you very much for this very nice talk. Other questions? I have a question by Rose Etier that I will read for you. Very nice talk, important work. Is transports through nuclear pores different in cage nuclei? Could be useful for cell transfection.
Yeah, so that's a very interesting question. I don't know. We haven't had the time to study this aspect, but for sure to kind of go into this direction. Things that we're also interested in is to understand if the chromatin organization is different in the cage and uncaged nuclei, because we believe that this deformation potentially is like modifying the structure of the chromatin and nucleus. And so, yeah, why not? It could probably influence the nuclear transport. And yeah, so full of possibilities in that direction also. But we haven't done experiments with this.
There is a question by Clément Campillo.
Thank you for the very nice talk. I have a question about the AFM experiments.
Yes.
So I just wanted to know how you imagine you could discriminate nuclear mechanics on cells that are such differently shaped. And I'm asking because we have similar questions in the lab. So could you comment a bit on that?
Yeah, so that's the big challenge for this AFM experiment that we actually are doing right now. So our problem is not so much on the cell shape. So the idea is that with the nuclear dye, we are really able to target and probe only the nuclear area. The only thing that we cannot really be sure is how much we are probing the nucleus and what is the participation of the cell cytoskeleton above the nucleus. One argument that kind of still validates this approach, where we don't isolate the nucleus, is that the cells are very flat. And so there is like a very thin layer of membrane and cytoskeleton above the nucleus. And so we believe that by indenting enough, we're still able to get the influence of the nucleus. But anyway, also having like information on the mechanical properties of the cell itself, or at least the nuclear area, will be like very interesting.
Okay, thank you. I'll be very curious to see the results soon.
Yeah, but actually maybe I'll come and discuss with you on AFM because it's a tricky experiment.
My pleasure.
Very good. By the way, I was surprised that you could have double caged nuclei.
Yes.
So it means nuclei are very big.
Yeah, so this nuclei are big, and on top of that, the surprise is that they are very deformable. So yeah, they are really able to envelop the groove completely, which is also always surprising to us. And on this aspect, that's why also we want now to test the behavior also of other cell types on the microgroup system to kind of understand if this is something like specific to the endothelial cells or not.
Okay, thank you. Okay, are there other questions? I don't see any. No. Okay, so thank you, Claire. So this is the end of this session about the rates of the SEED grants program. I think we can thank all the speakers because the talks were very good. And also I think we can congratulate them for having this nice grant. And we are expecting the future results. So now, and we are just on time, yes, we can proceed to the institutional part of this meeting here. As Abdul told you, Eric Labaye was supposed to come and speak a little bit, but he could not make it. And so it will be a video. So Sylvia, you take care of this?
It's Solange that has the videos. Here we are.
Okay.
Good afternoon, everyone. It's a pleasure for me to attend the second edition of the Biomedical Engineering Symposium, 1 year and a half after the first one in 2019. This special event demonstrates that the researchers of our ecosystem are more committed than ever to help improve our understanding and our mastery of this growing scientific field. I want to thank first and foremost Abdul Barakat, CNRS Director of Research at the LADIX and AXA Professor at École Polytechnique. I am truly grateful for Abdul's dedication throughout the years to making sure our research and innovations will be deployed for the benefit of society. He was therefore able to unite around the project of the Interdisciplinary Center of Biomedical Engineering all the researchers involved in biomedical research within the Research Center of École Polytechnique and then of Institut Polytechnique de Paris. Stemming from his project, Abdul's action made it possible to launch our Biomedical Engineering Seed Grant Program, which he leads, bringing to the next level the cooperation between researchers and doctors necessary to deploy feasible solutions to improve public health. This program reasserts the determination of Institut Polytechnique de Paris to play our part in the ongoing revolution for personalized, connected, and targeted therapies. This ambitious seed grant program could not have gone as far as it did without the generous support of Fondation Bettencourt-Schuller. I would like to thank them warmly, and in particular, Antoine de Boissière, its General Secretary, present today. Thank you as well to the Fondation of École Polytechnique for being a valuable partner on this seed grant program, as they are for many other enterprises that we put in place at École Polytechnique or Institut Polytechnique de Paris, with the intent to improve our society. This year, 3 research projects are rewarded, and I want to congratulate them personally. Clément Campillo and Elsa Labrune, Cédric Bouzig and Bruno Lina, and Abdul Barakat and Catherine Coirot for their innovative projects. The labs of École Polytechnique and Institut Polytechnique de Paris have an extensive knowledge in biomedical engineering, covering a wide array of activities in these fields. These include activities in biophysics and biomechanics, microscopic and macroscopic imaging, biomaterials, biosensors, and biomedical devices, just to name a few. Our campus also has strengths in fields such as artificial intelligence and data science that will contribute considerably to the evolution of medicine. As I mentioned earlier, because we believe that our research in physical science and engineering can make a significant contribution to medicine, we will soon be launching a biomedical engineering interdisciplinary research center. The goal for this center is to be a model at the national level for interdisciplinary science with extensive collaborations with the medical community and with other partners. Among important partners to build this interdisciplinary and inter-institutional center will be a number of hospitals, which I would like to thank as well today. Among them, Hôpital Marie-Lannelongue, Institut Curie, Pitié-Salpêtrière, Percy, Gustave Roussy, and APHP, represented today by Professor Alexandre Mebaza. A key partner with whom we will co-develop this interdisciplinary research center is INRIA. As you may know, INRIA and École Polytechnique have already a number of ongoing collaborations with the INRIA Saclay Center located here on campus, with joint research teams in LEAKS, CEMAP, and LMS. Moreover, we recently strengthened our partnership with the creation of the INRIA Center of Institut Polytechnique de Paris, including on health issues. I would like to finish on another major step we achieved in our ambition to improve our knowledge and innovation in biomedical engineering. We are proud to launch a couple of months ago, in partnership with the CNRS, with which we have strong ties, the BERTIP Graduate School, winner of the title École Universitaire de Recherche. The EUR structure, the teaching offering in BME at the master's and doctoral levels, and will integrate research into teaching and training at all academic levels. This program encompasses a highly selective 5-year integrated master PhD BME graduate program, the first of its kind in France, which emphasizes interdisciplinary training and provides ample opportunities for our students to interact directly with the research and medical community and the biomedical innovation sector. Among others, our students will benefit from a proximity with the Marie-Lannelongue Hospital, which will give them the opportunity to observe medical practice from the inside. We all look forward to seeing how the BERTIP program will evolve in the coming months and years, and how it will train our excellent students to become among the best innovators in biomedical engineering. To all, thank you once again for your presence today, which is a proof of your confidence and commitment to these exciting projects we have been building together for many years with École Polytechnique researchers and professors, and for 2 years now with Institut Polytechnique de Paris. Thank you all for your attention, and I look forward to the development of these exciting BME projects.
Okay, so thank you, Mr. President. And now I'm pleased to welcome Armand de Boissière, who is the General Secretary of the Bettencourt Schuller Foundation. And as you know, this foundation has given an invaluable support to this program. So, Monsieur de Boissière.
Thank you. Hello, everybody. So good to see you. I was very much impressed by all the previous presentations and speeches, and that's actually a great pleasure. I just will have a few words, not too long, and in French, because so far we are a French foundation. Voilà, donc en toute simplicité, quelques mots sur le programme et puis sur notre fondation. Je ne sais pas si Laura Ferry est dans le quartier, mais en fait, si nous sommes en relation heureuse dans ce programme, c'est parce que Laura Ferry-Fioni a présenté, lorsqu'elle travaillait à la Fondation, ce programme et que nous l'avons découvert et que nous avons décidé de le suivre. Donc, merci à Laura. Je voulais vous dire quelques mots sur la fondation. Vous savez, peut-être que c'est une fondation qui a une double particularité. C'est à la fois une fondation d'utilité publique avec des représentants de l'État, des personnalités qualifiées. Ça veut dire mener des programmes d'intérêt général dans un but non lucratif, mais en même temps, elle a un caractère familial très fort puisque c'est Liliane Bettencourt, son mari et sa fille qui l'ont créée, qui la dotent et qui sont les uniques financeurs et qui ont évidemment un rôle important dans cette fondation. Alors, il se trouve que dans cette famille, ça date du fondateur de cette famille, Eugène Schweller, qui était ingénieur de l'école de chimie de Paris. Ils ont vraiment une culture liée aux brevets, aux ingénieurs et aux chercheurs. Ils sont très liés, extrêmement sensibles. Ça nous aide et du coup, ça se traduit par le fait que la Fondation a, depuis sa création en 87, un peu plus de la moitié des fonds, à peu près 700 millions d'euros qui ont été attribués. Plus de la moitié correspondent à des projets de recherche. Et ce qui est assez caractéristique chez nous par rapport à d'autres familles fortunées. François, est-ce que vous m'entendez? Parce qu'en fait, je ne vois personne et c'est un peu compliqué. C'est bon?
On vous entend très bien.
Vous m'entendez, voilà. Bon, vous me coupez le micro si je déraille trop. Hein. Voilà. Et donc, la particularité de notre politique dans le domaine de la science, c'est que cette famille, de façon assez originale, cherche à financer de la recherche fondamentale, très en amont, vraiment de la connaissance de savoir qui sont mis après, comme vous le savez, parce que c'est votre métier à tous, je ne vais pas vous l'apprendre, à être partagés à travers des revues scientifiques à comité de lecture, dans des séminaires comme celui-là, gratuitement à disposition de tous. Ces connaissances fondamentales, peut-être qu'un jour, dans 20-30 ans, elles peuvent donner lieu à une mise sur le marché d'un médicament, mais vraiment, c'est relativement rare. Voilà. Elle intervient aussi dans deux autres secteurs qui sont la culture, avec le soutien à métiers d'art et au chant choral. Et puis dans le domaine social, nous avons une activité assez soutenue dans la lutte contre le handicap, l'illettrisme, et pour favoriser l'insertion des jeunes, notamment dans les quartiers. Alors, dans le domaine scientifique, en quelques mots, on a, je pense, finalement, ce n'est pas par hasard qu'on s'est rapproché de l'école polytechnique. Et en même temps, ce n'était pas du tout inscrit. Alors, pourquoi ce paradoxe? Alors, ce n'était pas du tout inscrit parce que, encore une fois, dans la recherche très fondamentale et pas forcément dans les sciences de l'ingénieur, nous ne soutenons pas les mathématiques, la physique, la chimie. Mais en revanche, depuis 2006, nous avons aidé un de vos collègues et camarades qui s'appelle François Tadei a créé la première école doctorale interdisciplinaire, qui est l'ED 474 Frontières du Vivant. Et à l'époque, ça a été assez compliqué, c'est-à-dire d'expliquer qu'il pouvait y avoir de la valeur à avoir des projets transdisciplinaires. C'était considéré comme vouloir rassembler des gens qui étaient médiocres dans leur discipline, qui n'étaient pas assez bons pour être chercheurs dans une monodiscipline et que tout ça n'irait pas très loin, et que c'était des bricoleurs. Voilà. Et il s'est trouvé que nous avons financé parmi les étudiants de l'école doctorale et du master des jeunes étudiants assez insolents qui ont été les premiers participants français au concours d'IGEM, que vous connaissez peut-être, voilà, de biologie synthétique. Et ils ont été les premiers à faire exactement ce que vous faites, du biomedical engineering, par exemple, en mettant des fonctions sur des bactéries qui libéraient des médicaments dans le ventre, en récupérant les fonctions d'horloge de certaines bactéries. Voilà. Et ces jeunes étudiants ont été très étonnés de voir que quelqu'un que vous connaissez peut-être, comme Alain Fischer et d'autres personnes comme ça, ont trouvé que c'était vraiment super. Et c'est venu par des étudiants de master en 2006-2007. Voilà. Donc nous, on a développé des formations interdisciplinaires. Dans notre chaire au Collège de France, on a eu aussi un de vos collègues qui s'appelait Gérard Berry, qui a lancé la première chaire qui s'appelait « Comment le monde devient numérique ?» Et puis après, ça a suivi par Matthias Fink, qui a fait une chaire sur les ondes, sur le renversement du temps, les ondes ultrasonores. Donc nous, ça nous a aidé à faire un pas de côté pour passer de la recherche pure en biologie sur une recherche interdisciplinaire et aux interfaces des sciences de la vie. Voilà. Et puis, nous avons travaillé avec l'INSERM, et l'INSERM nous a dit dans ces années-là que eh bien, depuis 10 ans, il n'y avait pas un seul médecin qui avait été reçu au concours de chercheurs statutaires à l'INSERM. Et que ça posait un vrai problème parce que les médecins qui étaient hospitaliers n'étaient pas assez solidement formés lors des essais ou des intuitions qu'ils pouvaient avoir, ce qui fait que quand ils faisaient des essais cliniques, ça pouvait être contesté d'un point de vue scientifique. Et puis ceux qui faisaient de la recherche très fondamentale revendiquaient une liberté qui était formidable, à laquelle nous sommes très attachés, mais qui pouvait être très éloignée de l'amélioration de la santé humaine. Voilà. Et c'est pour ça que nous avons co-créé avec l'INSERM l'école de l'INSERM Liliane Bettencourt, qui a déjà formé à peu près 340 étudiants en médecine avec un concours très sélectif où on va les accompagner pendant à peu près 14 ans pour qu'ils aient un double diplôme de MD-PhD. Alors, il peut y avoir une thèse précoce ou une thèse après l'internat, mais c'est vraiment intéressant et la période récente a montré que, comme ce que vous êtes en train de faire, mais la formation solide et précoce de médecins à la science est l'un des facteurs d'avenir. Donc, jusqu'en 2034, on a l'objectif d'en former à peu près 500. Et il se trouve qu'en fait, ils sont comme vous, ils sont tous super bons. Donc, jusqu'à présent, on est content. Et puis, il y a 3 ans, on a fait une évaluation de cette école et on s'est rendu compte qu'il manquait un truc, c'est-à-dire que quand ces médecins-chercheurs commencent une carrière hospitalière, il y en a quelques-uns parmi vous, eh bien, c'est très difficile parce que le système hospitalier fait qu'ils sont mangés par la salle. Voilà. Et donc, ils n'ont pas le temps, alors qu'ils ont l'énergie, les sujets de recherche, ils n'ont pas le temps de mener leur propre recherche. Voilà. Donc, on a créé un concours qui s'appelle les chefs de clinique assistants recherche, où on a décidé tous les ans d'ouvrir 4 postes soutenus pendant 4 ans, avec un budget par personne de 300 000 €, où nous rachetons le mi-temps de leur présence à l'hôpital pour qu'ils puissent créer leur propre équipe, un peu comme les ATIP à venir, avec des moyens qui sont mis pour qu'ils puissent maintenir leur recherche. Et ça, ça marche bien. Et on est très heureux. Et quand j'ai écouté lors des derniers comités d'orientation et de pilotage, et puis en vous écoutant maintenant, eh bien, d'une certaine façon, on voit que ça vous concerne ou qu'on est en plein dans la cible. Voilà. Alors, à côté de ces programmes, nous soutenons, j'ai parlé du Collège de France, on a créé une fondation qui s'appelle la Fondation pour l'audition, qui a créé un institut de l'audition, qui est le premier institut en France qui va faire de la recherche fondamentale et appliquée dans le domaine de l'audition, en fait, où on a copié le modèle de l'Institut de la Vision, que vous connaissez, certains parmi vous y travaillent. C'est vrai qu'à l'Institut de la Vision, j'ai la chance d'être administrateur de la Fondation Voir et Entendre. On est exactement souvent dans l'ingénierie biomédicale. Vous avez lu peut-être récemment une collaboration entre l'hôpital des 15-20, Pittsburgh, Bâle, 2 startups. l'université sur des projets en thérapie optogénique, sur des problèmes de rhinopathie. Voilà. Et là, il y a des procédés qui ont permis, au bout de 7 mois, à une personne d'arriver à revoir en noir et blanc, mais on est très proche de ce que vous faites aussi. Voilà. Alors évidemment, l'implant cochléaire, là aussi, on est dedans hein, pour l'audition, mais voilà. Donc, on a mis presque 120 millions d'euros dans ce projet pour essayer d'aller très vite et de faire la différence, et on a besoin de personnes comme vous, voilà, et d'équipes. Et le plus dur, c'est de trouver les personnes avec les projets et les collaborations entre les médecins, des chercheurs, des ingénieurs. Voilà. Et puis, vous savez aussi, on aide un certain nombre d'instituts. On ne cherche pas à aider les instituts, on cherche à aider les personnes et les équipes. Voilà. Donc, on aide des équipes à l'Institut Imagine, à l'ICM, à l'IGBMC, voilà, dans un certain nombre de domaines. Et puis, à côté de ça, on a des prix. Alors, pourquoi est-ce que je disais que c'est contradictoire? Je vous ai déjà dit qu'avec la politique de la Fondation, notre partenariat avec l'IX, c'est que, d'abord, ce n'était pas exactement de la recherche très fondamentale. Et puis, normalement, nous, on a l'habitude de temps long. C'est-à-dire que quand on soutient une équipe de recherche, c'est pour 5 ans. Ce n'est pas si facile à faire accepter par les temps qui courent, parce que vous êtes tous mathématiciens. Quand vous donnez 1,5 million à une équipe, immobilisée en revenus, pour avoir un revenu d'1,5 million par les temps qui courent, ça fait un sacré capital immobilisé dans votre dotation hein, pour pouvoir le servir. Donc, je ne vous explique pas les millions que ça représente. Voilà. Donc, notre budget, nous, de dons 2021, il est de 77 millions d'euros. Donc, ça veut dire un gros capital immobilisé. Et ça veut dire qu'on doit faire des choix par rapport à un certain nombre de projets. Alors, c'est contradictoire parce que 2 ans, franchement, il a fallu tout le génie d'Abdoul pour nous convaincre que ça allait marcher. Voilà. C'est pour ça que je ne voudrais pas vous rajouter de la pression sur vos épaules, mesdames et messieurs, les 6 binômes qui nous avaient présenté vos projets, en pleine période de Covid, avec le retard à l'allumage de tout ce qui doit être fait. Mais nous dire qu'en 2 ans, alors il a des mots magiques, hein, il nous dit high risk, high gain, tout ça, on espère que ce n'est pas des bonbons au caramel pour appâter le chaland, mais on l'a cru, et quand on vous écoute, on pense que ça va marcher, sachant qu'on n'est pas obligé de trouver un brevet au bout de 2 ans, hein, ni forcément un nouveau procédé. Si ça peut être des connaissances qui sont directement appliquées à l'amélioration de la santé humaine, on sera extrêmement heureux. Et voilà, on a été triste du fait que, vous le savez peut-être, ça a été dit avant que je n'arrive, que malheureusement, à cause des 3 confinements et des retards de travail dans les labos, dans les animaleries, la surcharge de travail dans les hôpitaux, ben on n'a pas pu faire l'appel à projet pour 2021 dans notre programme, c'est une année blanche. Donc franchement, ça ne nous réjouit pas. Mais donc nous, conformément à la souplesse de notre fondation, on a fait un avenant, on a prolongé avec nos amis de l'école et de la fondation de l'école polytechnique. Donc voilà, notre programme, il est prévu sur les 5 ans que ça soit 3+2, donc on va faire une évaluation des 3 premières années fin 2022. On a mis un petit budget exprès pour qu'il y ait à la fois une évaluation faite par le SAB interne du projet et de l'école polytechnique, et puis peut-être une évaluation externe pour avoir un double regard. Et je pense que c'est que bénéfique parce que forcément, les évaluations verront des choses améliorées, mais elles verront aussi la qualité de vos équipes. Ça donnera du crédit et peut-être une aide à l'école polytechnique pour créer l'institut dont le président a parlé tout à l'heure. Voilà. Donc, on est vigilant, on est plutôt très exigeant en termes de bonne gestion, de prévention des conflits d'intérêts, d'un certain nombre de questions éthiques et financières, mais globalement, pour l'instant, on est heureux et on est fiers. Et puis surtout, on dépend de vous, quoi. On est responsable des deniers qui nous sont confiés par cette famille et de l'argent qu'elle y met. Ils pourraient aller à l'étranger, ils ont choisi de rester en France, ils ont choisi la recherche, ils ont choisi l'école polytechnique et ce programme. Donc, On a un peu de pression, mais voilà, vos succès seront notre récompense, les amis, et on est confiants. Voilà, merci. Si vous avez des questions, je suis là pour ne pas y répondre.
Merci beaucoup pour tous ces encouragements. Laura, puisque tu es quelque part à l'origine de ce succès, tu veux peut-être dire un petit mot, te présenter?
Bonjour à tous. Attendez, je ne sais pas si je pense que ma caméra, je ne sais pas pourquoi je n'arrive pas à l'activer, c'est pas grave.
Ben si, c'est très grave, Laura. S'il n'y a pas de caméra à
Vous
l'école
voulez me voir,
polytechnique,
Armand?
s'il n'y a pas de caméra à l'école polytechnique, on arrête tout.
Je ne sais pas pourquoi elle ne veut pas s'allumer. Alors, bonjour à tous. Bonjour Armand. Et oui, effectivement, j'étais à la fondation quand ce projet a vu le jour. Je suis très contente, aussi bien pour la fondation que pour l'IP Paris, de voir que c'est un projet qui avance bien, qui apporte des projets et des sujets qui sont très intéressants. Et donc, j'attends de voir la suite, comme tout le monde. En tout cas, un grand merci à tous et bravo à tous.
Merci, Laura. Okay, so maybe we'll come back to English. Je vais quand même transmettre la remarque de Cécile Sykes. Cécile, tu peux peut-être le dire en direct. Sylvia, tu peux donner le micro à Cécile?
Non, je t'ai écrit le truc. Vas-y, lis ce que j'ai.
D'accord. Oui, donc c'était juste Cécile qui disait merci à Armand de Boissière pour ce discours sans phare, dans une transparence très anglo-saxonne que j'apprécie particulièrement. Et je pense qu'effectivement, ça traduit l'impression.
Nos actionnaires ont beau être propriétaires de L'Oréal, je ne me mets pas encore d'anti-rides, mais pourtant, j'en ai un stock. Ni de produits. Merci pour la remarque, c'est sympa, merci beaucoup. Ben, c'est comme ça qu'on travaille à la Fondation. Hein. En fait, on est un peu à votre service, on va dire.
Merci beaucoup. Okay, so I propose we continue with this very nice program. And to finish this day, we will have a long session by Professor Alexandre Mebaza. So Professor Mebaza is professor at the University of Paris, head of the Department d'Anesthésie et Réanimation at the Saint-Louis and Lariboisière hospitals, and also director of the INSER Mascot unit focused on the research of new biomarkers and biotherapies in acute disease. So Professor Mebaza has been practicing resuscitation for 30 years and wishes to establish new standards of care for this patient, but also for the survivors, because the recent pandemic has shown that it's not so easy. And in fact, intensive care has not succeeded in doing better than saving 1 out of 2 patients. So there is still a lot to do for that. And so I think it will be the subject of your lecture, Professor Mebaza. So I'll let you present. So I think you can share your screen. Okay, it's okay. We can see and it works. Yeah.
Thank you so much. Do you hear me?
And we can hear you, yes.
Great. Okay. Okay, then what did I do? Okay, sorry, let me come back to this. Just give me a minute. Okay, I think it should be good. Perfect, thank you so much. Thank you for the presentation and thank you for the invitation. I'm going to do give a lecture on the management of intensive care patients present and future, to try to show you what is our daily life and where really we need you in the future. And clearly we need to collaborate with scientists like you guys to try to go out from the Stone Age. I like this slide because you may feel it was so many centuries ago, but in fact, you will see in a minute that although intensive care units is the place where we have the most sophisticated techniques to try to save patients' life. And we used to say when patient enters in the ICU, one leg is in his bed and the other leg is not well located and looking towards death. We still do not understand what we are doing, and I'm going to show you some examples, and I will take the COVID as a main example. Today in the ICU, we really know nothing. And as I'm going to show you in a few minutes, we know a little bit, and at least those who are conscious that if we know something is really few informations, and we need to manage patients who are often have a risk, as you just said, of 50% to die in the ICU. And what we need in the future is try to understand really what is happening inside our patients. Let me show you some data on COVID-19. I guess every citizen in the world is expert in COVID-19, maybe a little bit less in severe COVID. And I'm going to show you how this disease that was on the TV screen for more than a year now, and where, again, we heard so many things. When we come back to the ICU, that was, as you remember, a limiting factor in the healthcare system, pushing governments to lock down all the countries, or many countries. When those patients were admitted in the ICU, in fact, we had only 3 tools that were recommended by all international societies. One is named the prone position. Then we are even not talking about medication. You will see what we are talking about, a prone position. The recommendation says when a patient is admitted in the ICU with hypoxemia, we recommend to give anticoagulants, and you will see that this started in my ICU and we discovered by chance. And the third thing is also a medication, corticosteroids, which is one of the oldest medication we have on our shelves. Then you see here, we are talking about a pandemic all around the world, and the treatment is very primitive. Then what is this prone position? Then a few years ago, you will see a French team showed that it may help. In fact, what we do is we put the patient on his belly, you see, as you can see here. And the reason why we do so is when we are all sleeping during the night, and when the patient is admitted in the ICU, and indeed he's staying in his back, the alveoli here on the top of his chest are opened, and the oxygen and CO2 can go through the alveoli and the bronchi. However, the alveoli that are here on the back close to the bed are closed. Why? Because we have the weight of the lungs, and then the alveoli are closed and cannot be open because, again, of the weight of the chest and of the lungs. And this reason why there was this idea of putting the patient in a prone position, which is not easy, we will see in a minute, but more importantly, the face would go to the bed, then we can have many issue on the face of the patient, even on the chest. But the big advantage is you see here that the area that were posterior and totally occluded, when we switch the patients, those areas are open, and then we have a lung that is open both in the ventral side and in the dorsal side. Then basically you see here that only half of the lung is ventilated, the other one is occluded, and here we can have the whole lung that is ventilated, again, getting oxygen and having CO2 being removed. Now, believe it or not, in ICU today, there are very, very few tools that are saving lives. We are getting millions of people every year in all ICU all around the world, coming from many diseases, going from a very severe acute myocardial infarction, very severe infection, intoxication, trauma, etc., etc. Almost the only treatment that is shown to save lives is prone position. It's even not a therapy. It's nothing magic. It's just putting the patient in a prone position. And when this patient came out in 2013, it was the first therapy that saves lives in ICU. And since that, there were almost no other therapies. And here is the figure that every medical student, every resident knows by heart, compared to the patients who stayed only in supine position. The patient in the prone position had a lower mortality, and you see that the mortality at day 28 was really markedly reduced, because we are talking about reducing by half the risk of death. Now, how do we do this prone position? And in fact, all the COVID patients all around the world, when they were admitted in the ICUs, around 5 to 8, 9 p.m., all the teams were putting those patients in prone position for the whole night. And in the morning, the fresh team were coming and putting patients back to the supine. And then prone position was roughly between 12 and 16 hours a day. Now, as you remember, many patients in COVID were obese because it was a risk factor. We still do not understand why it was a risk factor. Then I'm going to slowly try to show you how difficult it was to manage those patients with COVID. Then it seems to be easy to put the patient in prone position. However, many of them, as we said, were obese, and you already see how many people we need to put around. And you see that we go slowly for those obese patients, and it may take sometimes close to an hour. And in this patient here, we have even not too much sophisticated techniques. But this patient here, it's not COVID, because you will see here the healthcare providers were not wearing protective clothes. We will see in a minute how it looks with COVID. But you see here, there is only an intubation and mechanical ventilation. And in COVID, patients were even more, had more catheters, and it was more difficult. And this is exactly what you saw in many videos and many pictures. And this is coming from my ICU. Then you see here, we have the patient. The patient is in prone position. And here we are going to put him back in supine. But what is very interesting here, you see that really we are talking about personalized medicine. You see here, we have only one patient, and you have 7 healthcare providers. You have one physician, which is always a senior physician, because you see that there are already many catheters, and if one of them is disconnected, we can have a severe health issues. You will see 2 nurses and 2 assistant nurse, and 1 resident and 1 medical student. And here you maybe understand slowly what I'm going to talk about later on, which is how difficult psychologically it was difficult to manage those patients in the first wave of the COVID, because there was a fear of having people close to each other. We did not understand how this virus was going from one person to another. And we had those patients coming with COVID. And you see, despite this, and though those people were protected as much as they can, you see that 7 people here, just to save this patient life, twice a day for several weeks, they're accepted to be one close to the other one, and have a high risk of contamination. And I will show you and tell you later on how it was difficult for the healthcare providers this period, and how psychologically we have now to face big issues with healthcare providers who work so in a fear environment in that period. Now, as I told you, this is a really personalized medicine, because you see here one patient with 7 people taking care of him. And you see this is indeed totally different. And the ratio is different from what we can see with in airplanes with 2 persons taking care of 500 lives. And you see that really in the ICU, we need to have a team. And this is again during the first wave, where you see that the team has to be really working all together. And I can tell you during that period, there were really no hierarchy. Any physician, any professor, any nurse, any assistant nurse, when we had sometimes for 20 people to do a prone positioning between, let's say, 5 p.m. and 10 p.m., one after the other one. And then there were no social differences, and we had to do it, and we did it. And this was very, very helpful for our patients. I told you, treatment number 1 for a very severe COVID is prone position with all the advantages and all the risks. The 2 other medications that are now recommended is anticoagulation and corticosteroids. Anticoagulation came really by chance. And what do we mean by chance? In fact, when the first patients arrived in Paris, and when the first patients arrived in my ICU, I was lucky enough to have this case of a lady that was not intubated, obese, like many of the patients in the first wave, and she was hypoxic. And when we did the CT scan, we were surprised to see this pulmonary embolism. Then here you have this is a CT scan. Here you have the lungs. Here you have what we name the pulmonary artery with the blood flowing here in this direction. And you see here that the blood does not flow anymore because there is a big thrombus that is here. And we can see it also here. And this lady happened to come the day after we had 2 young persons, the age of 35 and 40, who died from a pulmonary embolism. And when we got those 3 cases, I could really not believe that it was by chance. And with my fellows, we went to look on PubMed. And I can tell you the 26th, 27th, 28th of March, on PubMed, when you put pulmonary embolism and COVID, the answer was 0 article. There was really nothing. And then together with the other intensive care of my hospitals, we decided to really quickly look on the legs and on the belly. And in fact, we could very quickly publish this paper on a severe, which was one of the first paper, maybe the first paper in a journal of cardiology, where we showed that 50%, it's a series of around 60 patients, and you see here that almost half of the patients in the ICU had a thrombus inside the legs and inside the veins. And we could also show a biomarker named D-dimer that is now used all around the world, and show that in those patients who had those clots, those thrombuses, the D-dimer were higher than the patients who did not have a clot. And what we mean about clot, here you have the leg, here you have the muscles. The vessel goes from here to here, and inside the vessel you see this big thrombus, and usually in the usual life you can have something that has 1 or 2 centimeters. Here we had sometimes 8 to 10 centimeters, and we understand that when this clot goes to the heart, it may block the circulation. But after this discovery, to try to see whether not only in the ICU these clots are present, we try to immediately see whether this is a health care issue in the Paris region and surrounding area, or whether it's only this issue is only located to it's only seen in the most severe patients with COVID. And this study was done by the Hôpital Georges Pompidou and Xavier Jouven, and he could show in his series, he could show that the patients with sudden death, some patients are walking in the street and they do suddenly sudden death. this patient with a sudden death, they could see that during the Paris lockdown in 2020, you see here, there was a huge increase in the patient's lockdown compared to the other years. And then immediately I contacted Bertrand Lude, who was the president of the forensic medicine in Paris. And he examined by CT scan all patients with a sudden cardiac death in Paris and surrounding. And the big surprise was that what we saw in the ICU is in fact true in the overall population. And we could see, again, this paper came out very, very early after the second wave. We could show that the pulmonary embolism was present in patients doing a sudden death anywhere in Paris, including patients of 20-ish years old. And we compared this to the year 2019, and the number of pulmonary embolism of during the 2 weeks of lockdown, or the peak of lockdown in 2020, in March 2020, March and April, there were as many pulmonary embolism as the whole year 2019, and we could see also deep vein thrombosis. Then, as I told you, anticoagulation started from here to go all around the world, but it was really, we discovered this really by chance because I had 2 or 3 patients coming together with a pulmonary embolism. The third medication that is I would say even maybe the only clear recommended medications in all the guidelines all around the world is corticosteroids. And you know that the trials was run in the UK. And in fact, they tested several arms, and the arm that came positive rather quickly was the arm that got dexamethasone, the oldest medication that we had, and all the other sophisticated medications going against that cytokines or other pathway didn't work. And this is reason why today we are just left with those medications. I would like to show you a clinical case to tell you behind the numbers there are really patients, and I will show you later that behind the numbers there are also healthcare providers. This patient came during the first wave, and we are still following him now. 65 years old, typical patient of the first wave. Comorbidities are known. No, he didn't travel. He arrived the 25th of March in our emergency room with anosmia. Unfortunately, they sent him back home because at that time it was really still, this disease was still unclear. He came back 4 days later. He was here getting more symptoms, and I remind you that at that time corticosteroids was not proven, anticoagulants was not proven. And on March 30, the patients came for the 3rd time. Really, you have to remember the issue that we had, and really our knowledge on COVID was really very, very few at that time. And the patient had to come back 3 times to the emergency room at the end to be accepted in the ICU. You see here that his oxygen saturation, that for all of us is 100%, it was 55%, basically close to the cardiac arrest. And as you know, this disease is terrible because we have what we name in French this hypoxémie heureuse, which are those patients who are coming with a huge hypoxemia but no tachypnea, and it's still unclear what is the mechanism behind this, but this is really was a terrible lack of sign for many physicians, thinking that the patients were doing well, although he was very, very severe. Then what parameters did we have to manage those patients? We had heart rate, blood pressure, a little bit of electroencephalography, oxygen saturation, mechanical ventilation, biology, and medical imaging. And really, as I told you earlier, very, very few parameters to try to fight this virus and the damages that were following this virus infection. What we try to do in these patients, again, is something very simple. It's just to help the body and patient's body to try to fight against this virus. And the only thing that we did for almost all the patients until recently, is to try to maintain tissue perfusion as much as we can, to try to have adequate oxygenation, to have a good oxygenation of the lung and good compliance. And most importantly, and this is for me was really the most important, is try to avoid doing mistakes, because you can quickly in this situation try to invent ideas and invent things. And really from day one until today, I really refuse to give any medication that was not supported by data. Then what did we miss? What did we miss for during all those waves? We miss to predict the degradation, and I will show you that we had in some patients, including this one, we had some signs, but we could not detect them. And I'm sure working with you guys, we can learn much more. What we may have helped us would have been to model the biophysical changes. And again, I will show you a case in a minute. And as I will tell you at the end, we need your advice, your expertise to try to understand how to integrate all those parameters. And maybe more importantly, when those patients with COVID were coming, we knew that there is risk factors. We knew obesity, we knew diabetes, we knew hypertension. But until today, we still do not know whether there were phenotypes of disease that were linked to that or that comorbidities. And here again, we couldn't do it because we are again on the stone age of managing those patients. Then this patient scenario is rather interesting, because when you look here on the oxygen saturation, we gave him a lot of oxygen. And here, because remember he came with 55, we gave him a lot of oxygen in the beginning, but unfortunately, you see here, it's 1 p.m., 2 p.m., etc. The patient very rapidly had a worsening lung and respiratory dysfunction. Then 15 liters per minute of oxygen. And you see here, after 1 hour, we had to give even more oxygen to achieve really a lower oxygen saturation. And in fact, I try to. You see here, I'm sorry, for some reason the slide does not move on. Yes, then you see here we had to increase even more oxygen to achieve a lower oxygen saturation, and you see that unfortunately the patient become worse and worse, and we had even to put the patient on mechanical ventilation here. Then you saw that he came with 55, we gave him oxygen, we felt that it went rather well, but he was degrading his circulation. But most importantly, see here that we missed here an example of, we missed here, you see this decrease in oxygen saturation. And if we knew that this patient, if we could interpret this decrease in oxygen saturation, maybe we would have intubated the patient much earlier. And it's very likely that this decrease in oxygen saturation that nobody could interpret today, if we had an analysis of a big database, we would interpret this situation much better. And here again, when we put the patient on prone position, awake, he had an improvement in oxygen saturation, and we were satisfied. But in fact, we were totally wrong, because a few minutes later, the patient did even a worse mechanical saturation. Now, even on mechanical saturation, you see that the patient was not doing so well. See here, oxygen saturation seems good. And then we inflated more the alveoli. By inflating the alveoli, we had the feeling that the patient was improving oxygen saturation. And then the resident who felt that increasing the pressure inside the lungs that was beneficial. If it happened once, we can do it twice. After 4 hours, he did it again. And here, for an unknown reason, the same maneuver in fact, led to a deterioration of oxygen saturation. Then again, just to show you how difficult it was, and this was only day 2, and those patients were intubated and ventilated for more than a month, then it was very, very difficult to follow those patients. And if we look on the chest X-ray, we had exactly the same scenario, a little bit of edema in the beginning, and we had more edema during the end. Interestingly, we could, at the end of the day, we put a tracheostomy in this patient in day 26, and we could extubate the patient. You see there is no intubation here at day 55. Interestingly also, and we don't know still, we cannot explain this, 3 months later the patients had a full recovery with the lungs, at least on the CT scan, that looks totally normal. But what is one of the area working on right now in COVID, but also we did it for all the ICU patients. This patient did the post-COVID morbidities. He stayed 1 full month in the rehabilitation unit, and we are still following him today, and he still did not recover almost 18 months after leaving the ICU. We still he is not moving properly. He still has arrhythmia in the heart. He's not breathing properly, etc., etc. Then this is really a major issue. In the future research in the ICU, maybe 3 areas where we could collaborate, and 3 areas where we may really help each other. And I heard interdisciplinary research earlier, and I think this sounds really nice to me. It's to integrate organ support. Those patients has a lung disease, they have a kidney disease, they have a liver disease, and integrating organ support would be nice. Integrate parameters, and I would like to, for a few minutes, talk about healthcare providers. Today, for this kind of patients, you see that there are different machines. Here we have a dialysis to try to support the heart. We have here an ECMO to support the lung and the heart, and we have also a mechanical ventilators to support the lung. And now industry is helping us to try to put all those devices in a single device. And there was a paper recently showing that the extra corporeal lung, renal, and metabolic support could be integrated in one machine. But maybe more importantly, we need to better analyze what we already have. And this is a picture I took 2 days ago in one of my patients. And you see here, this is exactly what the resident, what the senior physician is seeing every day. Then there are curves with different colors. There are many, many numbers. And I can tell you, there is no physicians who can integrate all those numbers. And in fact, when you look carefully on those curves, you see that the curves are working in silo. You have here electrocardiogram, that is, you see, it's a curve that is going alone. We have here oxygen saturation showing the oxygenation of the blood, but here you have also another curve. You have blood pressure. You can even, if you see carefully, you can even see that the curves are not simultaneous. You see there is a shift between this curve and this curve. And more importantly, all the physicians cannot integrate all those parameters, and every curve is really independent, and there is no link between the curves, and this is indeed totally wrong. See here the blood pressure, here the red curve. Here you have the blood pressure in the last hour. You see that in the last 30 to 40 minutes, the blood pressure seems to be, let's say, same level, and here you don't know why the blood pressure is decreasing. Indeed, there is no alarm, because the alarm runs only or rings only when the blood pressure is really very low. But here we are totally lost, because maybe this blood pressure means something. Maybe one of the parameters here, if it would have been linked to this blood pressure, would have given some indications. And really what we need is to integrate those parameters, but most importantly, to tell us some actions to do, and not only parameters, and having the physicians integrating these parameters to try to see whether there is an action to be made. And to try to integrate those parameters, we have a great collaboration with INRIA since 5 years, with Dominique Chapelle, who is running from INRIA side the collaboration. As you know, Dominique Chapelle is now the head of the Laboratoire Daniel Bernoulli, which is the APHP INRIA laboratory. And we got this grant named FHU Promise to try to markedly reduce the complication after surgery. I'm sure any of you knows a grand aunt, a grandmother that went to surgery and anesthesia and went out with some complication. In fact, it's very frequent. Here we have more than 12 million anesthesia in France. Most of those patients, when they go out of the recovery room, they have from the operating room, they had a full recovery, but we know that a large percent have a post-operative complication. And those complications lead to ICU stay. And as I told you, ICU stay, there is a disease for COVID is very high, it's 30, 40%, but in the for regular patients, it's already 20% death. 20% of the patients who are admitted in the ICU die during the ICU stay. But most importantly, our team demonstrated that even when the patients survive the ICU and go out of the ICU, if you follow them for 1 year, there is a 20% additional death. Then really, we are talking about patients who came for a surgery, sometimes a heavy surgery, but they go out either with some handicap or they die, and this is totally unfair. And recently in Lancet, a few years ago, it was shown that postoperative death is the third cause of mortality all around the world. In another paper, still in the same journal, they looked on the patients entering in the operating room all around Europe to try to see whether there are differences. And indeed, as you can see here, there is a huge heterogeneity for mortality for similar surgery all around Europe, with a Scandinavian country having a lower mortality compared to other countries. What is the reason why patients would die during the surgery or in the days or weeks after the surgery? There are two parameters that are increasingly studied, and this is hemodynamic parameters and brain parameters. Hemodynamic parameters, this is hypotension. During the surgery, sometimes the pressure goes down, and we try to restore this pressure. Sometimes we can do it quickly, sometimes not. And as you can see here on the y-axis, we have mean arterial pressure. And when the pressure goes down during the surgery, we have an increase in myocardial damage. And you see that this increase in damage is higher when the time of hypotension is longer, and this is also true for kidney injury. And we know in a French paper that if you can quickly restore blood pressure, you can reduce those perioperative complications. Another major complication of anesthesia and surgery is the delirium and cognitive disorders after surgery. And we know today that this is maybe due to the amount of anesthesia we are giving, but it is also known is if patients during the anesthesia, they got too much anesthesia, and we have signs that the EEG was flat, or that the patients had cognitive disorders when they came out from the operating room, this will not allow them to go back to work, and this is a major handicap. And then to try to limit as much as we can those complications during the surgery that have a mid and long-term change in outcome, we thought to try to set up some closed-loop automated system. Then today there are very few teams who try to have what I would name the first generation closed loop. Then you have here, let's say, the blood pressure. The anesthetist decide that there is a reference level on the beginning of the surgery. Then you see that blood pressure is moving during the anesthesia. And here at some stage, you see the blood pressure would go down. And in fact, today all around the world, we are working in reaction, meaning that the blood pressure goes down, we wait, we check, and then we give vasopressor to restore blood pressure. However, tomorrow, what we would like to do is to alarm the physician and anticipate 5 to 10 minutes earlier for the physician to give the vasopressor before the pressure goes down and try to prevent this hypotension. And the way we would like to do it, we would like to do it indeed for several parameters. And the way we would like to do it with Dominique Chapelle and his team is when we have the patient's data, we would like first to do a biophysical model, meaning that we look on the signals and we see when the signal change, and then do already a diagnosis. And today, thanks to the collaboration with some industry, we already collected with a very high high quality data of more than 3,000 patients in the operating room. And then the patient of the data can immediately be compared to all those parameters, and we will learn that that physician or that patient did that reaction, and this will give another signals. And by pooling those two signals, we can have this optimal diagnosis that will be sent to the physician, telling him, you will get a hypertension in 5 to 10 minutes, and we advise you to do this. And by this system, we can learn more. And we put together INRIA on one side, and the other side, we put together many centers from Assistance Publique, and the total is more than 200,000 anesthesia per year, which is really a big data set. And we are putting also a patients association together with us. I would like to finish with I told you earlier, the healthcare providers. We have today ethical issues and healthcare provider issue. Ethical issue is that in the ICU, we are teaching our resident and students to be technicians. When you see that science, you do this. We give a lot of procedures. However, we still do not know how to manage end of life. The decision to stop or limit therapies, there is no procedure, there is no technicity behind. During the pandemic, we were really lost because suddenly the institution decided that there should be no family visit. And suddenly we were discovering that the family was very, very important for us to manage our patients. And suddenly without family, we did not know how to handle those patients. And sometimes we had horrible situations with patients that left home, come to the ICU, died, and the family could not even see them because of the viruses, and they had to be buried immediately. Artificial intelligence is also putting some challenges in our daily practice, because we need to guarantee that the patients and the family understand what is behind the artificial intelligence, and organ donor is also an issue. As I told you earlier, we have a major issue with healthcare providers, and we have now a tendency since several weeks now all around France to see nurses leaving the ICU because we use them for years as technicians, and we are discovering, because the fear they had during the first wave, where many of the nurses and assistant nurses sent their families away and stayed alone to try to fight this virus, because there were a lot of ethical issues. We discovered that indeed the patient is vulnerable, but the healthcare providers are very, very vulnerable as well. In summary, intensive care units gets the more difficult patients in the healthcare system. However, I hope I convince you that today we are still in the Stone Age. Why? Because the data are generated in silo. You see that you have several curves, but the curves are not talking to each other. And worse than that, the treatment is also giving in silo. You see one machine close to the other one, and we need a double integration, having the treatment systems talking to each other, and all those treatment systems talking to the signals generated by the patients. And we should overall indeed save the patients, but we should protect. And I think really this is something that we learned a lot during the COVID, to try to protect the healthcare providers. Thank you so much.
Okay, thank you very much, Professor Membazar. It was a fascinating talk, and we have really been immersed in intensive care unit. Thank you very much for this and for all the prospects you have. So other questions in the audience? Right now I see nothing. Nobody. Oh, Sylvia.
Yeah, I have a curiosity. So you're developing the virtual physician assistant. So which are the main difficulties from a medical point of view to develop it?
To develop what? Sorry, I didn't hear the
The
first.
virtual physician assistant.
Ah, right. Well, we name it the virtual physician assistant to say that because the patients are not ready, the society is not ready to have a totally automated system. And it's true that in the beginning of our thought 5 or 6 years ago, we thought we could do like an airplane that takes off or land automatically. And today, the patients are not ready, the family of the patients are not ready, and they would like to have a system that helps the physician, but not a system that takes over the physician. And this is why we put together this virtual physician assistant system that may have, and this is the project that we are putting together, the RHU, that can help physician by 2 ways. One, by alerting the physician 5 minutes, 10 minutes, 15 minutes before the catastrophe that something would happen. And we know today, thanks to the collaboration with INRIA since 5 years, that by analyzing the signals more carefully, the same signals, we are not doing something, there is no rocket science here, just analyzing more carefully the signals and putting them together, having one signal integrated to another signal, we can achieve 5 to 10 minutes before hypotension, for example, we can tell the physician, be careful, here something is happening. But made more importantly, until now, when the physician in the operating room was seeing a hypotension, he had 3 or 4 treatment, and he would take almost one treatment by chance. And here, not only we are going to tell him in 5 minutes something is going to happen, but we advise you to use treatment A because the analysis of the curves are saying this. And then this virtual assistant, the challenge is to integrate those curves and then test on many patients and see slowly that the models that we are putting in place would be as close as possible from the 100% assurance that what the software would have predicted really happened.
Thank you.
Are there other questions? I just was wondering, you said that because of COVID, there were a lot of sudden deaths, and you don't have your, yeah. So I suppose that those deaths were not taken into account in the count.
The deaths were not taken into account for what, please? I
mean, for the official records, if there were sudden deaths in the street or whatever.
Yeah, indeed. Oh no, you couldn't believe the data. We know today that all what you see every day in your iPhone is far below what is the reality. Yeah, we know that, and in the first wave, you know that people showed that in fact all the old people who died in the non-hospital institution were not counted. But we know that many, maybe less in France than in the other countries for sure. But yeah, it's definitely not the total. Yeah.
Mm-hmm. Thank you. So, are there anybody wanting to put a question to Professor Mebaza?
I guess they are satisfied.
Okay, yeah, it was really fascinating talk. Thank you very much. Okay, so I think it's time now to conclude this day.
Thank you.
So first, thank you very much, Professor Mebaza. And I would like also to thank all the speakers of today, because it was a really great day. And these talks were all very clear and very interesting and very exciting, because this symposium really reflects the great diversity of the biomedical engineering at Paris. And for sure, there will be many more coming in the next years because of all the collaborations which we can put because of this institute. Just before leaving, I would like to extend a special thanks to Silvia Corsini, because indeed it was, I know, quite a big work to set up this program, and I really think that this day was a success. So thank you very much, Silvia. And I hope we will meet again next year, but face-to-face meeting and not through Zoom. So I wish you now a nice evening, and I say see you next year in reality. Goodbye. Sylvia, maybe you want to conclude something?
No, I would like to thank you, and thanks all the speakers and the chairperson that participate today. And I hope that we can organize more activities in September in person. So starting from happy hours and seminars directly at the ECOL. So thank you for today and your presence here. Have a nice bye.
Thank you and goodbye.
Bye-bye.